Okay, so I think everybody has just been admitted. So it's good morning from me and welcome to the first of the East Genomics showcases for 2024. And this will be presenting the work of the East Genomic Medicine Service and recent advances in the area of cancer genomics. My name is Professor Dick Sandford. I'm the Clinical Director of the East Genomic Medicine Service Alliance. So many of you will be familiar with the Genomic Medicine Service, and some of you, I assume, will have also been ordering genomic tests. But to briefly summarise for everybody what the NHS Genomic Medicine Service looks like. It was established in 2018 and it's been formed across 7 large geographies within NHS England. And you can see the East geography in pale blue here. It covers the East of England and East Midlands. And the Genomic Medicine Service comprises the genomic medicine alliances and also the laboratory hub. So together they comprise the Genomic Medicine Service. And the GMSs are working to ensure equitable access for eligible patients to a nationally commissioned genomic test directory. And that really is designed to be as inclusive as possible. So it should not matter where individuals live or which hospital they are treated at. They should all be able to access genomic testing. And it's supporting the establishment of testing pathways for a very, very wide range of diseases. We're obviously talking about cancer today, but we'll be talking about other treatment pathways in the 2 additional showcases this week. And a major part of the work of the alliances is supporting the NHS workforce and clinicians in requesting genomic tests. And through all of that work, introducing new models of care that support early access to genomic testing and hence improved high quality personalised treatments. Next slide, please. And this is our geography in a little bit more detail. All of this information is on the East Genomics website, but just so that you can see, the East GMS covers a large population of about 11 million people. There are 29 NHS trusts, acute trusts, mental health trusts and community trusts in this particular area, and 11 integrated care boards providing care. And we also have 3 genomic laboratories the hub based in Cambridge and additional laboratories in Leicester and Nottingham. Next slide, please. So I just very briefly wanted just to show you this particular diagram, and this tells you a little bit about the structure of the East Genomic Medicine Service. So we have an integrated governance structure, and what you're really going to hear a little bit about today is those aspects of our work delivered by the so-called working groups. And we are obviously focusing on cancer today, but we have working groups covering nursing and midwifery, medical and rare disease, pharmacy, the clinical genomic services, research and innovation, information and digital, and comms, as well as education and training. And all of those workforces report into a structure that ultimately reports into our East Genomics Executive Board, which is
Mondays or 60 hours.
Which is chaired by, sorry, we just need some microphones off, which is chaired by one of our partner trust chief executives. And obviously included in all of that is the input from our strategic partners, which includes industry, academia, primary care, and the cancer alliances, from which you're obviously going to hear a little bit more about today. Next slide, please. And critical to our work, not only for delivering our work, but the co-design of our work, is our patient and public voice panel. And this is chaired by Chris Hines and Eddie Blair. And this panel is very, very active in our work. It's represented across all of our working groups. And these and members of our PPV panel bring a range of their own experiences from genetic testing and their own established networks. So they're key to how we operate. And again, you'll hear a little bit more about this today and also in the coming days, the additional showcases. Over the last few years, and the Genomic Medicine Service has been active since 2018, but we've moved from the delivery of what we would call transformation projects into developing a series of networks, genomic networks of excellence. And again, you'll hear about activity of these networks, some activity of these networks today, and they cover a whole range of different areas where genomics is going to have a very substantial impact in the coming years. So they cover prenatal medicine, in cancer, circulating tumour biomarkers, which you'll hear today, hemato-oncology, again, you'll hear more about today, rare and inherited disease in another showcase this week, cardiovascular disease, artificial intelligence, and also pharmacogenomics, which you'll again hear woven into all of our showcases. And more recently, the development of an infectious disease network of excellence where genomics is going to have a big impact in terms of pathogen sequencing. So there are some very, very exciting developments coming. Next slide, please. And this is just a list of some of our strategic partners. And if you represent an organization today where you feel that obviously you can wish to participate in the genomic medicine service, can make an additional contribution, we would very, very much like to hear from you. So please get in touch. Next slide, please. And I've already mentioned our East Genomics website. Here it is. Please go there to contact us and gain further information. And all of our resources are there and available to you. So we look forward to hearing from you via our website. And next slide, please. And finally, I'm not going to go through the agenda for today. It's a packed agenda covering a wide range of subjects relevant to cancer genomics. We are going to finish with a question and answer session chaired by Emmanuel Nkube this afternoon, but also just to highlight, we've got 2 additional showcases in rare and inherited conditions on the 10th, and also for prenatal medicine on the 11th.
I'll introduce the first speaker of today's session. The session is How is Genomics Impacting Cancer? This will be presented by Prof Srinivasan Madhusudan, our cancer lead at East Genomics. Madhu is Professor of Medical Oncology at University of Nottingham. His clinical and translational research expertise is in breast, upper GI, and ovarian cancers. Madhu's preclinical lab research is focused on the evaluation of DNA damage, signaling, and repair in cancer. He is the director of the Nottingham Ovarian Cancer Research Centre, based in the Biodiscovery Institute, University of Nottingham, and joined East GMSA as the East Midlands Cancer Lead earlier this year. So over to you, Madhu.
Excellent. Thank you, Emmanuel, Richard, and Ian, and good morning to everyone. So Emmanuel has given me a very challenging talk. Over 20 minutes, I need to convince you as to how genomics is impacting cancer. So Ian, over to the next slide. So what I was planning to do over the next 15-20 minutes is, this is a diverse audience. Some of us are clinicians, some of us are non-clinicians, some of us are, patient reps and so on. So what I want to do is really set the scene for this morning as to what is it that we're dealing with and what's the problem to hand and how are we going to address this in the genomics medicine, East Genomics and the East Midlands Cancer Alliance and the East Anglia Cancer Alliances. So to do that, I think it'll be useful for us to think about backgrounds to cancer genomics. The historical perspectives to cancer genomics is quite fascinating, and it's a rapidly evolved field over the last 10 to 15 years. So we'll spend a few minutes talking about that. And then I wanted to give you an insight of, what are all the services that we are offering within the genomic service, in various situations when patients unfortunately land up in a cancer center, treatment for cancers, and also their families. So what are the various services that are available? And of course, the Genomic Medicine Service Alliance has to work in close collaboration with the East Midlands Cancer Alliances or the East Anglia Cancer Alliance. I think this is critical for us to achieve what we have set ourselves towards over the next couple of years or so. So I'll give you a flavor of what is the strategy that we have come up with that we'll work towards over the next 2 years. And then I want to give you a snapshot of an audit that we have done through the EMCA, Vicky Kiesel leading on it, and a GMSA audit on DPYD testing, which has really kick-started what is it that we want to get. We want to understand what's going on here in the genomic service in this part of the world, and what are the future directions. So that's how I've kind of designed over the next 15-20 minutes. We'll very much welcome any questions at the end. So Ian, next slide, please. So here is a timeline of what's been going on in this field. We all know, in fact, all of us here in this audience know that cancer is disease of the genome. Yeah, so there are various hallmarks of cancer that have also been updated. But underlying all this is a basic fundamental difference or alterations in the genome that has led to a cancerous phenotype. So we all agree that cancer is a disease of the genome. So genome is what? DNA and DNA sequences. So if you look at the history of how the genomics has evolved over the years, it's quite fascinating. The Sanger sequencing started in 1977. Probably many of us in this audience were not even born at the time. So Sanger sequencing in 1977. Look at the Human Cancer Genome Project, the Human Genome Project. And we may all remember that Tony Blair presents in the TV, 2003, 1990 to 2003, Human Genome Project. 2006, the Cancer Genome Project comes along. And then, of course, over this period, the technologies that lead to high resolutions of sequencings have changed. You had the first generation Sanger sequencing, then you had the second generation sequencing, then you had the third generation sequencing. Now in 2016, we've also sequenced circulating tumor DNA of over 15,000 patients. And there's a lot of talks coming up around circulating tumor DNA later this morning. 2016, circulating tumor DNA. Meta-analysis of all cancer genomes, 2,658 cancer genomes from 38 cancer types. So if you look at this, from 2003 to 2018, over 15 years, there's been a rapid progress in genomic evaluations of cancer. And not surprisingly, now that's become mainstream, because much of the companion diagnostics for cancer therapeutics involves some type of genomic testings. Either it is whole genome or a targeted gene sequencing of some type. So it's become critical for us, for those of us in the clinical space, treating patients with cancer, getting the genomic testing has become critical part of, in fact, a daily part, bread and butter of what we do in oncology. So to complement that, as Richard mentioned, NHS came up with the genomic testing service, and we have 7 GLHs and 7 GMSAs spread across the country. And then we have the East Genomic Laboratory Hub and the Genomic Medicine Alliance. It's quite a big catchment area of 29 hospitals, 29 NHS trusts offering this service to patients. So that's the grand scheme of how historically the genomic service has evolved in the UK. Next slide, Ian. So when you look at a cancer patient, how do patients land up in a cancer center in a situation where they need genomic testing? So our patients are coming to a cancer center through different routes, either population screening, breast cancer screening, colorectal screening, and so on, or a primary care presentation to the GP, prompts a 2-week wait referral. Patients eventually get into a secondary care. Various investigations happen, and then a cancer diagnosis is made. So at that time is when you think about, do I have to do a germline testing or a somatic genomic testing in this situation when I have a patient in front of me? So that's how you have a broad classification, germline testing or somatic testing. Germline testing, and we'll talk about it in a few minutes. The germline testing, of course, helps not only the management of future cancer risks of patients and their relatives, but now we have precision oncology therapeutics for certain germline mutations that again we'll talk about in a few minutes. Then, of course, you have the somatic tumors. Okay? In somatic tumors and also in certain germline tumors, you have the tumor, you want to know the genomic phenotype of the tumor, because as I told you before, companion diagnostics are leading to precision oncology treatments for patients. So this has become an essential part of medical oncology care. You are prescribing targeted therapies based on genomic analysis of tumors. So that treatment, not only we know is improving survival outcomes for patients, but we are also able to give some prognosis. We can also give prognostications for individual patients. We can also do cancer surveillance after they've had their precision treatments. And if they have a recurrence of some type, then again, they go through the genomic testing for precision oncology strategies. So this is how the oncology in the genomic era has completely changed. 10 years ago, it was an entirely different way of operating. And now we've changed how medical oncology practice happens in patients. So coming back to the germline mutations and somatic mutations, let's think about it very briefly. In germline mutation, one of the alleles already got a cancer, already got a mutation. So you got one mutation that's already happened, and then you need to have a second mutation, and then subsequent mutations, which eventually lead to a malignant phenotype. In somatic tumors, you have a normal cell that acquires first mutation, second mutation, third mutation, and so on, eventually leading to a malignant phenotype. So you have the germline and the somatic. So in the medical oncology space, the most common tumors that we see is the sporadic tumors, okay, which have somatic mutations. Hereditary cancers and familial cancers is still a rare part of the oncology practice, but nevertheless, it's a significant aspect to it. Up to 10% of cancers can be hereditary. Ian, next slide, please. So germline testing, does it save lives? Absolutely, yes. And I've just taken two of the commonly investigated genomic testings in certain tumors. We are all very familiar with BRCA1 and BRCA2, PALB2 as well. So BRCA1 and BRCA2, without going into the details, we know that germline mutations in these genes, tumor suppressor genes involved in DNA damage signaling and response, can increase the risk of breast cancers, ovarian cancers, male breast cancers, prostate cancers, pancreatic cancers, and certain communities are particularly prone to have these kind of germline mutations, particularly the Jewish population. PALB mutation, germline mutations, and it's 1 in 750 people are carriers of this. Okay, particularly triple negative breast cancer. Patients who carry this are prone for triple negative breast cancers. There's a risk of ovarian cancer, male breast cancer, and pancreatic cancers. So this is a classic example of how a genomic service and a genomic testing, if it picks up a germline mutations, you know that these are all the cancers that can happen. Next slide, please, Ian. So how does it affect patients? So if you have a patient in front of you who is known to have a BRCA testing, it impacts significantly because it alters the surgical recommendations that you may want to give to this particular patient. Instead of a wide local excision for a breast tumor, perhaps you want to give them a much more radical surgery options like bilateral mastectomy. If somebody carries a Lynch syndrome, we'll come to it in a bit, you may want to offer colectomy. Risk assessments for future cancers. If somebody has breast cancer, they'll be prone for ovarian cancers. If somebody has a Lynch syndrome and colorectal cancer, they are prone for gynecological cancer. So it has an impact of future assessments. And you may be able to do some chemotherapeutic chemoprophylaxis for some of these patients, risk reduction strategies with drugs such as tamoxifen, and of course, personalized chemotherapy plan. And this is an exciting area of cancer therapeutics where you use PARP inhibitors, or we know that some of these tumors that carry BRCA deficiency can be platinum chemotherapy sensitive. Okay, so it does help you plan treatments in various ways. So it does have a major impact on patients and their families. Next slide, please, Ian. If, for example, you have a healthy BRCA carrier, what happens to that patient? Your screening strategy is different. You probably do an MRI scan. You will do an MRI scan in patients in their 30s to 50s. Okay? Mammogram in 40 to 70. You will offer them preventive surgery such as mastectomy or salpingo oophorectomy. So it has a major impact on lives of patients. Next slide, Ian. So Lynch syndrome. So this is an interesting problem at this point. So Lynch syndrome in the UK, about 175,000 people in the UK have Lynch syndrome, and surprisingly, a large proportion of whom are walking around without being aware of this. It leads to about 1,100 colorectal cancers in the UK. And it is the most common cause of inherited colorectal cancers and endometrial cancers. The proportion may be less, 2 to 3%, but nevertheless still significant. And there are other tumors that patients who carry these germline mutations are prone to. Example, gastric cancers, prostate cancers, pancreatic cancers. Ian, next slide. And very similar to patients who carry BRCA deficiency, Lynch syndrome has a protocol for diagnosis. All endometrial and colorectal cancers will have somatic testing, preferably on a biopsy. It'll probably be an immunohistochemistry or look for microsatellite instability. And then there are protocols around if all the 4 proteins are present, it's unlikely to be a Lynch syndrome. 50% of sporadic tumors may show an absence of MLH1 and PMS2, but it needs further testing with methylation. And then the high-risk patterns prompting germline testing. So this is a protocol that's available for patients with colorectal cancer where you want to look for Lynch syndrome. Next slide, Ewan. And again, like BRCA, it impacts. If a patient has Lynch syndrome, needs a colectomy. And for therapeutics perspective, if somebody has Lynch syndrome, we know that they dramatically respond to immunotherapy. They have long-term complete responses and partial responses and survival outcome improvements if they carry Lynch, the genes that are prone, put patients at risk of Lynch syndrome. So from a therapeutic perspective, it becomes super important that we understand in patients with, for example, GI oncology area. Next slide, Ian. What about carriers? You want to give them more colonoscopy. And here is an interesting point, chemo prevention from aspirin. And we know that we are not doing that for patients in this part of the world. There are not many patients receiving aspirin, not in the carriers receiving aspirin, despite knowing that they have a Lynch syndrome phenotype. So this is a major challenge for us as to how do we bring forth these kind of chemo preventative strategies in this population. H. pylori detection, so on, and you know, women with this lesion, with this germline mutation, probably needs a radical surgery, yeah, and men may need PSA testing over the age of 40. So huge implications for carriers for Lynch syndrome. Next line, please. What about for somatic tumors? As I told you before, you know, majority of our patients are somatic tumors who come to a cancer center. And here we are, and Richard mentioned that briefly to all of us in his introductory slides. National Genomic Testing Directory for Cancer. You know, a big chunk of this is dedicated towards sporadic tumors. And look at this figure. Every organ from which tumors can arise, there is a genomic testing that is currently available. Yeah, brain cancer, head and neck cancer, sarcoma and melanoma, and everything else in between, we have genomic testing. Next slide, please. So how are we doing in the East Midlands? I probably have 1 or 2 minutes to run through these slides. So one of the things that we want to do is a big genomic strategy here, and this is a work in progress. And we have our joint working with GMSA and EMCA joint strategy over the next 2 years, where our first aim is to have audits and capture what are we doing right and where do we need to make improvements, areas around research and around education. Okay, next slide. Ian. So for the EMCA audit, I just want to highlight over in 10-20 seconds, thanks to Vicky Kiesel for sharing this slide. So one of the key aspects that we have done recently is to look at mainstreaming of the colorectal, endometrial, ovarian, and breast prostate tumors. You know, how are we requesting this genomic testing? Is it a mainstreaming through the MDTs, or are the patients being referred to the genetic services? And we had to send surveys to 7 trusts and understand what's happening here. Ian, next slide. And what we have found here is that all trusts would like to support with mainstreaming, and we had to do some progress with Derby. 5 of the 7 trusts have a genomic strategy. Mainstreaming is happening using many models, either through CNSs, oncologists, or surgeons, genetics practitioners, family history services. So we may need some fine tuning of the service. There are huge gaps in mainstreaming, family history clinics, family history screening as well. Next slide. So that's from the germline testing for families. Okay. What about in sporadic tumors? And I just want to give you another 30-second snapshot of a key audit that we have done around DYPD testings. So in medical oncology, it's a big challenge for us in the sense that there are many patients who receive 5-FU based chemotherapy. And although 85% of the chemotherapy drugs get catabolized through the enzyme called dihydropyrimidine dehydrogenases, or DPYD as we call it. About 5% are the proportion of these chemotherapy drugs that actually go into tumor cells and kill the tumor cells. So if you have a germline problem with DPYD, where there is an excessive presence of the chemotherapy drug in the system, it can lead to undue toxicity. But the key question for us here has been, despite routine DYPD testing that's been around for about 2 to 3 years, data on variant prevalence across different tumors, ethnic groups, and impact of social deprivation on access to test are limited. And we have used this as a backbone for us to understand how we can roll this out to other genomic testing service or other genomic tests in solid tumors. Next slide, Ian. So here we captured about 1,500 patients. That's all patients who were referred to Nottingham University Hospital for this test. Okay, next slide, Ian. So here is the top headlines of this particular work involving 1,500 patients over 3 years. When you look at ethnicity, more than 80% of patients to whom we are doing the DPYD testings are Caucasians. Afro-Caribbean, just around 1.3%, Asian 3%, others 1.6%. And it was also giving us another challenge. Ethnicity data is not widely captured. In fact, almost 13% of our patients, we don't know what their ethnicity is. But nevertheless, Caucasian seems to be having a lot of DYPD testing. Look at the tumor types. Again, colorectal is the highest numbers where we're doing the DPYD, followed by breast and then other tumors. Ian, next slide. And when you look at the DPYD variants, this is really interesting. The published literature says somewhere in the region of 4.9 to 6.5 thereabouts %. And we see around that figure, about 103 mutants, we have, not mutants, variants that we have captured for our population, about 7%. 7% is the detection rate. Okay. And again, the HAP-B3 is the commonest variant that we detect that is very much in concurrence with the published data. Now, here is the interesting data. When you look at DPYD mutations across ethnicity, again, Caucasians, we seem to be picking up more of the DYPD, the DPYD variants in Caucasian compared to Afro-Caribbean or Asian population. And within these various ethnic groups, colorectal cancer again stands out. So what's interesting here is colorectal cancer, Caucasian seems to have high incidence of DPYD. Yeah. Next slide. And the next question, of course, is, and this is, I think, is a key question, and we wanted to test a model system. Okay. Is the DPYD testing equitable across socioeconomic backgrounds? And to address this question, we have worked with our collaborators in the various faculties, Faculty of Engineering, Department of Computer Science, and the Digestive Diseases Center with Guru Prasad Aital and Phil Quinlan, and Torrance Simple, a PhD student in Graziela's laboratory, to try and address this question. Next slide, please. So don't want to go into the details of it. Basically, what we looked at is DYPD testing rate and matched it with index of multiple deprivation. Yeah, and there are specific parameters that we can use called lower super output area analysis. The deprivation index looks at income, employment, health deprivation and disability, educational skills, training, crime, barriers to housing and services and living environments. And you put that in this equation to do this IMD analysis. Next slide, Ian. Yeah, so this is the headlines here that I just wanted to share, which is actually a pretty reassuring data. So this block here, A3, this is LSOAs with the high DYPD testing rate, but are on the most deprived third of all of LSAOAs in Nottingham. So what that is clearly telling us is there is no visible difference between the most and the least deprived areas in Nottingham with regards to DYPD testing. And again, giving us a clear indication that this is a Departmental of Health mandated test. And every patient who is deemed suitable for this chemotherapy will have to have this test. And we are compliant, and it is an equitable distribution of these tests across all ethnic, socioeconomic backgrounds. Can we achieve similar kind of, equitable distributions for other genomic testing? So this is a big question that we are now going go and explore within our various audits that we have planned. So next slide. I'm in the last 2 slides here. So the conclusions here is GPYD is 7%, Caucasians and colorectal more common. We have not observed any variations in DYPD testing, which is quite reassuring. So here, next slide. What are the future directions for us in this in the East Genomics and the Cancer Medicine Alliance? I think we are very clear with regards to we need a joint strategy here, and that joint strategy has already kicked off, and we've got projects moving in this area. I've given you examples of 2 audits, and we are also focusing quite a lot on education. And from the initial analysis that Ian showed us, it does feel that lots of education needs to happen so that we request more and more of this testing. Next slide, Ian. And finally, I just want to introduce the East Midlands Oncology Genomics Roadshow, which has started already with myself and Vicky coordinating this across various leading cancer centers and teaching hospitals and district general hospitals. We have done this in Nottingham already, and next we are planned for Northampton, Kettering, Leicester, Derby, and Lincoln in that order to try and get folks engaged in this service and in this exciting area of oncology. Next slide. And that's my whistle-stop tour of the impact of oncology. Sorry, I might have run a little bit over time, but I hope it gives you a flavor of what's going on here. Over to you, Ian.
Thank you very much, Madhu, and grateful that you could compress such an important piece in that short space of time. The next session, pleased to introduce, is Lynch syndrome, past, present, and future, and this will be presented by Prof Ruth Armstrong, consultant in clinical genetics at the East Anglian Medical Genetics Service and Lynch syndrome lead for East GMSA. Dr. Anna Wilsden, local consultant clinical geneticist at Nottingham University. Welcome, Ruth and Anna.
Thank you very much for that introduction and for promoting me. I'm not actually a professor, but if we could have the next slide, please. Okay, so I'm very excited to be able to showcase Lynch syndrome, past, present and future. And we aim to give an overview of the condition and the many risk reduction interventions that are available if patients are appropriately identified. We will show how this has been achieved with the help of the National Lynch Syndrome Transformation Project, highlight areas that still require further work, and initiatives that hope to address this. Next slide, please. So by way of introduction, Lynch syndrome, as you've already heard, next slide, please, is an autosomal dominant condition that's adult onset, and it's due to pathogenic variants or what we previously knew as mutations in mismatch repair genes. And the 4 most common ones are MLH1, MSH2, MSH6, PMS2, and rarely EpCAM deletions. And it's a relatively common genetic disorder, as you've already heard, with 1 in 400 people being affected. And it's the most common cause of inherited colorectal cancer and endometrial cancer. And there's also an increased risk of other cancers, such as gastric, small bowel and pancreatic cancer. And the risk of actually developing cancer over a lifetime or up until the age of 75 is dependent on the specific gene, the type of cancer, and also on sex. So for example, a man with an MSH2 pathogenic variant may have a lifetime risk of colorectal cancer of over 65%. Next slide, please. So as the previous speaker alluded to, there's a number of different diagnostic pathways, and they usually start on the tumour, or ideally on the initial biopsy. And they include a combination of immunohistochemistry and MSI, and then depending on that initial result, they usually need a confirmatory blood test to identify the pathogenic germline variant. Next slide. So once we've confirmed Lynch syndrome in the patient, predictive genetic counselling, testing and risk reduction interventions are available for at-risk family members in adulthood. Next slide. And as already alluded to, this is extremely important because there are a number of simple risk reduction interventions that can reduce the risk of developing and dying from cancer. Simple healthy lifestyle advice such as increasing the amount of resistant starch in the diet can be helpful. Encouraging patients to be symptom aware and act promptly on any new symptoms. Some families may choose reproductive options when planning their families, and it's really important that patients know how to access testing for their at-risk family members. A simple aspirin a day can reduce the risk of colorectal cancer by 60%, and testing for and eradicating a common stomach infection can also reduce the risk of gastric cancer. Next slide, please. Colonoscopy bowel screening can reduce the incidence and mortality from bowel cancer by 50%, and there's also the option of risk-reducing surgery. Say, for example, a lady may wish to have prophylactic hysterectomy once she's completed her family. And immunotherapy has revolutionised the care of those with advanced cancer. Next slide, please. So if we reflect on the past to inform the future, Lynch syndrome has actually been around for a really long time. It was first described back in 1895 by a Dr. Walthen, who noted that a number of his seamstresses and female family members had all died from uterine cancer. And it was initially known as hereditary non-polyposis colonic cancer, or HNPCC for short, to differentiate it from familial adenomatous polyposis, which is a genetic condition where the bowel is carpeted in thousands of little polyps. It's now known or named after Dr. Henry Lynch, who first described two large families in America way back in the 60s. And then the first gene, MSH2, was discovered in 1993, and by 1995, all four had been identified. Next slide. So nearly 30 years later, there are around 200,000 people with Lynch syndrome in the UK, but 95% of people don't know they actually have it and therefore can't benefit from the risk reduction interventions. Next slide, please. Even when a family has been diagnosed with Lynch syndrome, they are not always appropriately investigated. I'm sure many of you will remember Stephen Sutton, who sadly died at the age of 19 from bowel cancer. He had a known family history of Lynch syndrome, but unfortunately his new symptoms weren't investigated promptly, and he documented this across social media. Next slide, please. So in 2017, a combination of stories such as Stephen's, poor case ascertainment, and general concerns that Lynch syndrome wasn't being recognised and investigated appropriately led to an expert group of clinicians, charities and patients recommending that there was a national registry of individuals with Lynch syndrome, and that fed into a quality assured surveillance programme with a dedicated clinical champion for Lynch syndrome in each MDT. NICE subsequently published their recommendation that all colorectal cancers and all endometrial cancers were tested for Lynch syndrome in 2017 and 2020 respectively. Next slide, please. Next slide. And NHS England produced this handbook to support local systems, and it basically portrays the different testing pathways. Next slide. Lynch syndrome was also included as one of the GMSA National Transformation Projects. Next slide. And the objective of the Transformation Project was to embed robust pathways to deliver universal Lynch syndrome testing for patients with colorectal and endometrial cancers in England through the Genomic Medicine Service Alliances. Next slide. The aims were to provide national and regional medical and nursing leadership, to deliver a comprehensive education package, to develop regional and national registries through the National Disease Registry Service, or NDRS, to support the National Bowel Cancer Screening Programme, the 2-yearly colonoscopy, to introduce mainstream germline genetic testing within the actual cancer MDT, rather than there being a referral to clinical genetics, to develop regional expert networks, and to provide performance monitoring and audit. So the project delivery involved a huge team, which was hosted by the 7 GMSAs across the country, and I'd like to take this opportunity to thank anybody who has been involved in that. Thank you. Next slide. So thinking about the present, the GMSA Transformation Project has been hugely successful in increasing the number of patients who receive initial testing for Lynch syndrome. So in the East GMSA back in 2019, around 45% of patients with a newly diagnosed colorectal cancer had IHC testing for Lynch syndrome. By 2023, this had increased to 96%. Back in 2019, only 17% of new endometrial cancers had testing for Lynch syndrome, and by last year that had increased to up to 95%. We've also been able to establish local registries in each of the regional clinical genetic services in Nottingham, Leicester and Cambridge, and then these feed into the national disease registry, and that in turn informs the National Bowel Cancer Screening Programme, where patients are automatically invited for 2-yearly colonoscopy bowel screening. And we've also introduced Lynch syndrome champions into each cancer MDT. We have delivered education to over 2,000 local patients and clinicians, and this has been multi-format. It's been online training resources, webinars, patient information events, which have been extremely well received, and we've also written to every patient that's on the disease registry. We've formed 2 expert Lynch syndrome networks, which Anna will expand on later. We've improved the IT infrastructure, introduced a patient app to facilitate follow-up, and Cambridge has also been involved in extension site testing for gastric cancer to see whether it's actually helpful to test all gastric cancers for Lynch syndrome as well, along with endometrial and bowel. But as with any project, unfortunately, there are still a number of outstanding issues. In the East GMSA, we have only been able to mainstream 21% of bowel cancer germline testing and 26% of endometrial cancer testing, which is below the national average. And there are a number of different capacity reasons for that. There are also significant delays in germline testing of up to 18 months, with an average turnaround time of 376 days. And this is mainly due to a lack of lab capacity due to a national lack of suitably qualified scientists. And we are working so hard to try and improve this, and we've begun to see the turnaround times coming down. There have also been some concerns regarding funding, and we need to be better with primary and secondary care liaison. So as the project drew to a close, we went out to Lynch Syndrome UK and conducted a national survey of patients and families with Lynch syndrome. And we received over 300 responses, but 42% of those were diagnosed prior to the project, pre-2017. One concerning factor that's already been picked up upon was that over 95% were white British, and therefore we do have concerns about equity of access. And the patient survey really picked up on the huge impact that a diagnosis of Lynch syndrome has, not only for the patient but also for their family. It picked up on a lack of clinician knowledge and awareness, and also the need for improved and coordinated care and a lack of psychosocial support.
Talk to you a bit about what we have in place now, in particular the expert networks. But as Ruth highlighted, there are these really key issues raised by the Lynch syndrome UK feedback. So I thought I'd look at what we can offer now in relation to those themes. So the first theme is lack of knowledge. And it's not uncommon to hear that not all clinicians know about Lynch syndrome, or perhaps they only know about the bowel side of it. This is really frustrating for patients with Lynch syndrome. And it's a struggle that they share with all individuals who are diagnosed with a rare disease, that they will become the expert, and they'll often know more than that health professional sat in front of them, which is a really big burden. But we can't expect every health professional to know about absolutely everything. So we've done some targeted teaching, providing resources, providing links to the expert networks, for example. And if you do want to find out more about Lynch syndrome, there are loads of educational resources on the East Genomics website for patients and for healthcare staff. So there's lots of different categories on there. There's recordings of educational sessions, there's handbooks, and there's template letters, for example. I'm not sure my slides has advanced, unfortunately. Can somebody just click? And again?
Just trying to now, Anna. Yeah.
Oh, okay, sorry. I might have started a bit too early. There should be another one to appear. Okay, and then if you go on to the next slide. Okay, fantastic. So quite a few of the experts who've been involved with providing this education and have taken on these Lynch syndrome roles have also spoken at events that we've arranged for both healthcare staff and patients who are interested in finding out more. So this was our last one in Nottingham, and I know that there are lots more events planned. Having a chance to hear the team for the bowel cancer screening programme talk was really helpful for me, because now I can talk to other individuals who I see with Lynch syndrome with much more knowledge. So it's been a really helpful experience, and that we've learned from each other too. There'll always be more people to educate, but we are making progress and we're making sure the resources are there. So the next big issue was the need to improve care. The feedback talked about more joined up care, knowing that their clinician can access the relevant knowledge and understand what's going on. So we set up the expert networks to facilitate this. So there's two in East Genomics. We've got the East of England and the East Midlands service. But how do they actually improve care? Well, part of their role is to provide quality and continuity of care for patients across the region, but they're also a source of knowledge and other resources that can be accessed by other clinicians. So essentially, it's a bit like a hotline to specialist expertise when it is needed. But what exactly is the expert network? So it's a central team that includes lots of specialties relevant to Lynch syndrome. The expert networks run a monthly MDT that clinicians can refer their patients to if there are any queries, or if there are complex problems. For example, do we need to consider whether that patient needs more extensive surgery because of their Lynch syndrome diagnosis, or would joint specialty operating be helpful? But it should also provide access to a Lynch syndrome specialist for patients who are concerned about symptoms, for example. So if you see an individual with Lynch syndrome and you aren't sure how to proceed, you could refer your patient to the local expert network. And there's lots more information on the East Genomics webpage about the expert networks and about how you can refer to them. So the next theme was the need for more support. Give patients the tools to make it easier for them to access the care that they need. So how are we tackling this? Well, the first way we're doing this is by providing information to patients. So as Ruth mentioned, we've written to lots of individuals with Lynch syndrome, and this might be notifying them of the expert networks, for example, or the latest research studies that we've opened, or by signposting them to the resources that are available on the East Genomics site, which includes all the recommended care for individuals with Lynch syndrome. The other thing that Ruth again has already mentioned is an app that's been developed by our colleague in Leicester, Dr. Julian Barwell. So he's been instrumental in developing that. And essentially this means that as an individual with Lynch syndrome, you can have something at your fingertips to show health professionals. And it's got a huge number of different roles. It can help you keep track of screening and which investigations you might consider. And I know Professor Barwell is going to speak to you more about this later, so I won't go into that any further. The third way we're trying to make it easier is by breaking down the barriers to care. So what we want to be able to do is translate the recommendations into that individual's care. There's some really clear guidance now about taking aspirin. We know it significantly reduces the lynch of cancer and Lynch syndrome, but what has become apparent is that despite us telling everybody we see with Lynch syndrome to strongly consider taking it, a good proportion probably aren't. And I'll talk to you about some of the barriers that we've identified when I come on to the future plans. But for now, there are some great resources out there, like this Decision Aid produced by NICE. In the East Midlands, we're really lucky to have a GP attached to the expert network, and she can chat to patients to help them decide if aspirin is right for them. So if you've got a patient with Lynch syndrome, or you are an individual with Lynch syndrome and you want to find out more, you can ask for a referral to the MDT to be able to sit down with a GP who understands Lynch syndrome, who also understands everything else in GP land, about whether aspirin is the right choice for you, for example. So the final theme from the Lynch syndrome UK feedback was recognition of the personal impact of being diagnosed with Lynch syndrome. And it's clear this is a huge unmet need. There have been lots of things put into place to make sure the screening and the physical care is improved, but we need to think about the impact of having a diagnosis of Lynch syndrome. So again, in the East Midlands, we're lucky to have a psychologist attached to the expert network there who has a background in cancer and specialist knowledge in Lynch syndrome. And we would encourage referrals to the expert network in East Midlands for individuals who you think might benefit from this service. I think generally there is a push to get more psychological help and services available to individuals who are diagnosed with cancer outside of Lynch syndrome as well. But in the meantime, if we can focus on improving the rest of their care and make life a bit easier for them, if they're not having to battle to access the care that is recommended, hopefully that alleviates one burden. So what does the future hold? Lots of exciting developments and opportunities, but this is going to need continued funding and hard work. So the Lynch registry in the portal is now up and running, which has really streamlined things. The fact that we now have an active register of patients in our clinical genetics department means we have an accessible list that we can use to inform patients about new care options. And the NDRS means we can see how individuals with Lynch syndrome do over time. And that's been a really successful project. And there are plans to set up similar registries for a lot of the other cancer predisposition genes too. I have the next slide, please. Okay, so as I've just talked a little bit there about registries and the success in this case, there are lots of other projects springing up too, which one of them includes the prospective Lynch syndrome database. So this is a tool where clinicians can upload information about patients and how they do to give us the really sort of more accurate figures about what the cancer risks are in Lynch syndrome, for example. And I cannot emphasize enough how valuable it has been to work more closely with our colleagues in colorectal surgery and gynecology, for example, as part of this work. But as I've mentioned already, we do need to expand our reach and work more closely with other specialties too. So in Nottingham, I think in particular, we need to look in more detail about translating the clinical recommendations into clinical care. So it's easy for me to say, you've got Lynch syndrome, I really strongly recommend you take aspirin. Nip along to your GP, have a chat to them about it. But for some, that's quite a complex decision, and it might not be a discussion that the GP feels confident to have. And the aspirin dose is currently an off-label indication. So there's quite a few roadblocks in the system that are now becoming apparent, and we need to address this. So in Nottingham, we're really lucky to have a fantastic interface with our primary care team, which I'm planning to make full use of soon, so people don't get stuck in at that point. We're looking at how else we can make it easier. So one of the complexities of, well, is working in hospitals, it's difficult to understand the complexities of GP working because we're completely different systems in some ways. So one of the issues that we picked up is how the diagnosis is coded on the notes and how you keep prominent issues like Lynch syndrome in view when you open up the record in GP practice. So Vicky Kiesler has been working really hard with teams in primary care to standardise a code and to make sure it is listed on that person's problem list and it doesn't just drop off. So it should always be there in view. On to research. So this is a really exciting area. Currently, there are quite a few studies opening and they have the potential to really change care again. So the first is preschools. This is open in Nottingham and I think just about to open in Cambridge. And it's focused on endometrial cancer prevention and quality of life. So we know that women with Lynch syndrome have a higher risk of endometrial cancer. So risk-reducing surgery to remove the womb, the fallopian tubes and the ovaries can be considered because it's 100% effective at preventing ovarian and endometrial cancer. But there's not been any good studies of quality of life after that surgery and very little research into quality of life in Lynch syndrome in general. So this study is aimed at all adult women in the UK with Lynch syndrome who've not had a gynecological cancer. And it's a short questionnaire. And the hope is that collating these results will mean that women with Lynch syndrome and their doctors can make an informed choice about whether surgery is right for them and what the effect would be on quality of life. Lynch Choices is a fantastic new website that's being developed through the University of Southampton, Cancer Research UK and CanVar, and it's going to provide all sorts of resources and decision aids. Currently, this is a testing version, so they're looking for feedback on this site, but it's potentially another great resource that will be able to signpost individuals with Lynch syndrome too in the future. And we've talked a lot about what you can do for endometrial cancer and colorectal cancer and the screening for that, but we haven't really concentrated so much on the other cancers, such as the cancers in the urinary tract, which are also a factor in Lynch syndrome. And the risk is not insignificant, and these cancers are potentially curable if you can find them at an early stage. So a very clever group at the University of Newcastle have designed an assay which essentially screens your urine for evidence of cancer in the urinary tract. So this test looks in the urine for fragments of DNA which have been shed. This is called cell-free DNA. So your DNA is normally kept safe in your cells, but sometimes little fragments are released and are floating around in things like your blood. So, for example, in a pregnant woman, we can take a sample of her blood and pick out the DNA that belongs to the baby. And it's a similar principle. So cancer cells divide and grow quickly. So there's lots of these fragments about. So handily, if you've got a cancer in the urinary tract, there'll be lots of these fragments of DNA in your urine, and they have a characteristic signal. So this test can look at these fragments to spot if there's a cancer there. And not only is this test good at detecting cancers, but the signal should also disappear when the cancer's been removed. And even better, this test uses urine samples that you can pop in the post. So they're looking at funding to try and get this set up. Disclaimer, obviously, they don't use tiny scissors to do this, but as I've been talking for a while, I thought I'd better put something a little bit less serious in. But hopefully this is a study that we can, hope to see in the near future. So obviously a lot of funding has gone into Lynch syndrome to get us to this point, and there have been some huge successes, but it's important we ensure these services are really cemented into place and adequately resourced. So the national programme of support is finished as of March this year. And while some parts of the pathways have really robust funding, others don't. And we're looking at ways to support these sections of the pathway. Lots of the relevant charities and professional bodies have banded together to support these requests for funding. So we're really hoping that this shows the funders how important it is to continue this work. So thank you to all of those charities. I think overall, we've massively overhauled the Lynch services and the future is bright. There are still gaps, and sometimes as clinicians, we can't see those so easily. We can audit how well we're doing testing cancers and making diagnoses, but if the patients feel unsupported, then we haven't got it quite right. If it feels like, you're trying to navigate spaghetti junction rather than a nice straight line of care, then we need to know. So I'd encourage any individuals with Lynch syndrome or any health professionals who are involved in their care to please let us know where the gaps are. So I'm going to finish there, but I'd just like to thank everybody who's been part of the national project, the staff that have taken on the challenge of setting up this service, the funders and all the charities, and of course, all the individuals with Lynch syndrome and their families who we work with. Thank you.
Thank you, Anna and Ruth. It segues quite nicely, actually. The next session is Finding the Fragments ctDNA. This will be presented by Dr. Brent O'Carrigan, who is a consultant medical oncologist at Cambridge University Hospitals, where he treats patients with melanoma and kidney cancer, as well as leading a portfolio of cancer clinical trials to improve the care of these patients. Currently, he's also leading the East Region rollout of the National Circulating Tumour DNA project. Welcome, Brent.
Great, thanks Emmanuel, and thanks all, and thanks for the excellent presentation before me. Really fantastic and a great segue, as Emmanuel said. Fantastic. Team, do I advance the slides myself there, or I can?
I'll be doing that.
Okay, great.
If you just let me know.
Yeah, great. Thanks so much, Ian. Look, thank you so much. Also, as Emmanuel said, we'll talk through now one specific application of the work in cell-free DNA or circulating tumour DNA that was alluded to in the previous presentation. That's in our actual National Lung Cancer Genomics ctDNA pilot program. So next slide, please, Ian. And so we'll just speak a little bit about molecular testing in lung cancer. As folks on the call may well be aware that lung cancer is quite a heterogeneous disease now. So in previous days, people have had lung cancer and really required chemotherapy. That's not the case anymore. Now there's really a wide variety of driving mutations found in patients with lung cancer. And there are different mutations I won't dig through in detail today, but really these are critically important because they can affect patients' care most immediately. Patients with a particular EGFR mutation, most commonly, they would receive actually tablet therapy that block the EGFR mutation rather than chemotherapy. And there's a whole long tail there of less frequent mutations, but again, some of these can be exquisitely sensitive to particular therapies instead of chemotherapy. So lung cancer really is the template child for really personalized medicine. The defining lung cancer and the active molecular pathways really impact patients' care, both in early and late stage disease. In early stage disease, for example, people now have curative surgery, and now they have additional preventive or adjuvant therapy, and that will vary either chemotherapy or tablet therapy driven by the presence or absence of molecular mutations in their lung cancer tissue. So next slide, please, Ian. And one of the ways we can detect that is by blood testing. So historically, really we would use tissue testing. This has been done before really on tissue biopsies, which means either a CT biopsy of a lung mass or perhaps a bronchoscopy and biopsy from a lung sample, and then molecular testing. And that just takes time. It takes time to book the procedure, takes time to analyze the histology, the tissue, and then further time again for the molecular diagnostics. And that's improving, but it still does take quite some time. As you can see here, what we're looking at now is that actually with some blood test markers, we can get a much quicker access and much quicker readout of those molecular testings for those particular patients to really get the right drug at the right time for those patients. So I will move on to the next slide there, please, Ian. And so what we're talking about with cell-free DNA, onto the next slide as well, is a novel assay you just heard about from the previous presentation, looking for tiny fragments or sheet fragments of cell-free DNA in the bloodstream. Now, it's a normal process, and you can measure DNA in your blood, today as normal body cells grow and die and change. And our previous speaker mentioned actually in women's care, so in pregnancy care, you can actually measure differences between the baby's DNA in mum's blood and then the mother's blood alone to really type out differences differences in any gene changes in that baby. But here in tumour DNA, we're talking about people with cancer, perhaps early or late stage cancer. And then as those cancer cells grow and divide and die, as they normally do, they release into the bloodstream tiny fragments of the tumour DNA. Onto the next slide, please, Ian. That can be picked up and measured and can be distinguished from the normal healthy DNA. And so what you have here is an exquisitely specific sample of tumour DNA, and you can measure that both in quantity over time, but also its characteristics. So what mutations does it carry? Can you measure different gene changes that are unique to the tumour and different to the patient? And there's a range of applications that you can, this may have a role in the future of detecting cancer early, as we're doing, for example, with screening CTs. Can you find cancer before it's apparent on scans or with somebody with symptoms? To monitor therapy, so if someone's had some therapy, can you see changes? Can you see a reduction in the amount of DNA in the bloodstream? And also changing therapy. Can you see different mutations, and would you change therapy based upon that? So there's a range of applications that this technology will probably have in cancer care moving forward, but the talk today is really about the current use in lung cancer. So on to the next slide, please, Ian. So specifically, and to the next slide again, please, Ian. This particular assay, again, we've mentioned how, historically this requires a tissue biopsy to really type out those gene changes, mutations, which can take time, can be quite invasive. The procedures themselves carry risks, whichever way you might want to access the tissue. But here, a blood draw, a simple blood draw, which can be done alongside your standard of care bloods in clinic, that can actually type out and accurately measure and quantify the gene changes, if they exist in the tumour, different to the patient's bloodstream. So on to the next slide, please, in there as well. So the particular assay that was the source of this path was actually initially a commercial assay from Guardant, so Guardant 360. And this simple blood draw takes a simple blood draw, so it's 2 blood tubes taken in your standard of care clinics that doesn't require refrigeration or specialized transport pathways. And then really the internal lab turnaround time is around 7 days from laboratory sample receipt to sample readout to really test and see if you can pull out the presence or absence of any mutations in that patient's bloodstream, and what those mutations are, the specific points in those mutations and how common they are, their frequency, what's the volume of metastasis of mutations in that patient's tumour as well. So while this began as a commercial assay, it's actually been brought internally now into the Royal Marsden and to the NHS for hoping to be usable care. So on to the next slide, please, Therein. So what we're talking about here is this particular panel. Actually, it's a broad-based panel and really measures around, I think, 74 or 75 different point mutations. So outside of lung cancer, the same assay, Guardant 360, has applications in a range of other tumour types, for example, colon, lung, breast, melanoma, and others. But at the moment, the most clinically applicable pathway is in lung cancer. And so this assay was chosen to be one of the commercial providers to meet the needs for the National Genomics ctDNA pilot. You can see in the green boxes there, this particular assay measures a range of other critical mutations in other tumour types, so EGFR in lung, HER2 in breast cancer, and others. But its application to lung cancer is the most specific pathway as well. And also, that's the assay as it stands today. One of the benefits of using these assay-based platforms is they can change with time. So at the moment, it's irrelevant for lung cancer, but it will be possible over time to use different mutations and apply this to other tumour types as well. To the next slide, please. And so most specifically in lung cancer, this is an extremely sensitive test. So here really, you can see the sensitivity boxes there in the pathway. And the most relevant box is the top one where it says single nucleotide variants. And that's because I guess the test is only as good as what you know you're measuring. And in lung cancer, most of the driving mutations or the clinically important gene changes are point mutations in a particular gene, for example, the EGFR gene. And so for that specific type of DNA change, which is what we're looking for in lung cancer, this test is very, very sensitive. And you can see there it really approaches, as long as there's sufficient DNA in the bloodstream, a really very high sensitivity. And that's important because we want these tests to be reliable, to be accurate, to have a quick turnaround time, but also be sensitive and robust so they can really inform clinical decisions in real time for patients as well. On to the next slide, please. Therein. So here what we're measuring is, while the test has been deployed across a range of tumour types, I mentioned that the panel can measure targets of interest for lung cancer, but also targets of interest for other tumour types as well. It has a very high rate of detection across all tumour types, but it's particularly sensitive for lung cancer. So both that means small cell and non-small lung cancer. And it's both a feature of the fact that mutations we see in those cancers are just more amenable to detection on these kinds of assays, so they're very highly applicable. And also they're clinically, the kinds of cancers cancer where this assay is really helpful. Often lung cancer is obviously in the lung or in the lymph nodes in the chest. They are difficult areas to biopsy. So not only is the test quite sensitive clinically, but it's also much more applicable. If people can have a blood draw rather than a deep biopsy, that would be very helpful for patients and get them the care they require sooner. On to the next slide, please. And so there's a range of trials that have shown that this is a helpful pathway. I'll just draw your attention to the bottom of the two blue plots there. So this is 280 patients actually in the US, comparing if they're having their diagnostic testing on standard of care tissue testing, which means a tissue procedure, biopsy, and then sampling, or with a blood draw concurrent to the usual pathway. And you can see they're getting their results really in 9 days. So that's much faster than the tissue pathway. And in that case, having not had to go through a procedure or a biopsy. On to the next slide, please, there, Ian. And also, most critically, we're looking to see this slide discussing how, sensitive and specific, what are the rates of false positives and false negatives. And what I want to draw your attention to is both, the yellow box here on the lower right. So here it's saying, this is looking at the false negative rate. If the person does not have a mutation, how reliably negative is that test? And it's reliably negative. So we rarely see false positives with this test. If that's negative, that's because there's not a mutation present or detectable in that pathway. So it's a test with very high, with a very high and very sensitive detection rate as well. I'll move on to the next slide, please, there, Ian. Yeah, that's a few more builds. And so critically, while here the laboratory turnaround time is 9 days in the lab, you need to include a few days for sample transfer. So at the moment, the program is running on a turnaround time of about 11 days total, which includes time from patient in clinic, blood draw, sent to the reporting laboratory, and results then back out to the clinical team. So really, you're getting results back in about 11 days total time from patient to clinical result, which is 9 days of lab turnaround time. And so because this assay is really, quite promising, quite effective, and quite fast, this has been chosen to be the basis for the national platform to roll out there as well. So to the next slide, please, Ian. And also it's shown to be helpful in lung cancer in a UK setting. So this trial is looking actually during the time of COVID in the UK. So it's a sample size of around, I think, 50 patients from the Royal Marsden, which informs, I guess, the UK context for the impetus and inspiration for this pilot. And so we said during COVID, as folks will recall, there was less access to tissue biopsy than even in our usual care. And so this paper looked into see how helpful was a blood draw in expediting patients' care when there were, in this case, unusually long delays for tissue diagnosis. And it really found that actually, that you can detect the majority of variants from those blood draws from patients early and quickly, and action care sooner. That's a critical point, that rather than just a result in somebody's inbox, that this materially changes patients' care. You're accessing care sooner, and also better, more personalized care sooner. I've mentioned that in the absence of any mutations, people have often chemotherapy, perhaps after surgery, or if they have advanced disease. But in the presence of a driving mutation, they'll have different and personalized therapy. This is getting patients the information to their clinicians and for shared decision making, sooner, but also tractable information that really changes what people do with patients in the clinic when they're there for those results as well. So the inspiration from this trial led to the first pilot of initially 700 patients in England. I'll move on to the next slide, please, Ian. So here we're now talking about how that trial has really pushed away and, I guess, provided the impetus and inspiration for the national GMSA lung cancer ctDNA pilot. This has been a phased rollout, and really here the long line of scientists say we know that these tests and these assays are clinically effective, but they're quite expensive. So this national pilot is looking to see, actually, are they deployable in real care? Do they materially change patients' care, and are they cost effective? So there's a lot of health economics built into this pilot. The pilot's going in 3 different phases. So the first phase was, I think, 700 patients, so 100 across the 7 genomic regions across the UK, and that's actually run from the commercial assay I mentioned. So Guardant Health was the lead provider in many, but I think not all of the laboratories, and that was successful and accrued very well and was very promising for patients. And so that's been brought into a phase 2 pilot, and that pilot was 2,000 patients. And from phase 2 on, we've now done a situation called a tech transfer, where the genomic laboratory hubs have brought the Guardant technology in-house, and that's now run inside the North Thames laboratory, so that's basically the Royal Marsden. When they're running really effectively, it's the guided assay, but internally to NHS level service specifications, protocols, SOPs, and delivery time. So this is now becoming an NHS in-house branded test to really roll this out for patients. So the phase 2 pilot was really looking at 2,000 patients to broaden and test that we can run that assay internally on NHS hubs, which was done quite successfully. And so now we're expecting to roll out to 10,000 patients nationally through 2023, 2024. The line of sight from that larger pilot really is actually NHS commissioning. So this whole endeavour is to say, can we prove the utility? Can you prove the cost effectiveness? Can we make the business case for this assay to be rolled out for usual care to patients across the UK in the lung cancer clinical pathways? On to the next slide, please, Ian. And so where that's running locally is across our East of England pathway. So we're doing across this pathway through both the East of England and the East Midlands GMSA, and we've got now a whole range of trusts on board. Myself and the team are really quite proud that we've actually made this a distributed pathway. This is not confined to university centres, but as you can see there, and this is expanding, a whole range of trusts across all of our geography are involved and actively accruing patients to this sample, which means that people can have, wherever they live across all those trusts, and others are more than welcome to join the pathway as well, access to this novel, innovative, currently quite expensive commercial private test for free, funded by the GMSA inside their usual care centre, and they can have that blood draw alongside their first clinic consultation, and they will have molecular results in their inbox with their clinician with about kind of 11 to 12 days, which can inform their care and change the care they receive on the next pathway. And so my team have worked very hard to make sure that we can roll this out across all the pathways and areas and into the trusts involved as well. And this is an open door. So we'd like this to be increasing in uptake and increasing in prevalence across the patch, because hopefully this will become routine commission testing towards the end of the year or perhaps Q1 2025. On to the next slide, please, Ian. So moving on to the next slide. So for trust to be involved, I actually recommend, if your trust isn't actually active, contact myself in the first instance to really connect up and bring the trust on board. But then actually, once your trust is an active site, really, you request test kits directly from the Royal Marsden laboratory. They will send out test kits to your site directly. On to the next slide, please. And it's a matter of a simple blood draw, and it's a matter of a blood draw from the patient. It does not require any dedicated consent either for data purposes or for clinical testing. This is now considered a secondary diagnostic test along with your usual biochemistry and routine samples. 2 tubes, simple blood draw alongside their routine testing, and the labels provided. It's then sent away across to the Marsden. To the next slide, please, Ian. And that's then sent away to the laboratory hub with the relevant request form, and then the results are then returned into the inbox of the requesting clinician with a copy to my laboratory team as well. On to the next slide, please, Ian. And it really is that pathway. So it's sent away through Royal Mail. There are different safe seal boxes, so there's samples can be tracked across the postal service and received in the laboratory internally. On to the next slide, please, Ian. And then results are back to your team within really about kind of 11 or 12 days. Over to the next slide, please, Ian. And so at the moment, people are having those. At the moment, the advice would really be, this is for people with suspected, so imaging suspected stage 3 or stage 4 lung cancer living anywhere in the East of England or the East Midlands, or actually across the whole region. So even if you're on the border trust, the adjoining GLHs will be your point of contact as well. And if your trusts are not involved but would wish to be, you can contact myself on the email there also, and we can ensure your trust is live and active and able to access this innovative test for patients in your area, in your geography nearby as well. So I can leave the final slide there for a moment, but I'm happy to take questions, comments from those on the call, on the floor, or just check on the chat to see if there's anything in the chat there, Emmanuel.
We were going to pick up questions at the end of the session, okay, Brent, but if there's any, we'll send directly to you to pick up, and then we'll wrap up after the sessions.
Yeah, great. Thanks, Emmanuel. So I'll have to probably actually jump up before the main questions, but I'm happy to be contacted now or at any time, and I can pick up questions offline as well. That's wonderful.
Brilliant. Thank you very much. Yeah. Thank you, Brent.
Thank you so much.
Our next session, Advances in Brain Cancer, will be led by Dr. Patrick Tarpey, who is lead scientist for Solid Cancer East Genomic Lab Hub and senior research scientist, Wellcome Trust Sanger Institute. Welcome, Patrick.
Thanks, Emmanuel. So good morning, everybody. What I'd like to do is talk about our aspiration to improve the outcome of clinical outcome for patients with brain cancer using insights gained from whole genome sequencing. So gliomas account for almost 30% of primary brain tumours, but 80% of malignant ones and are responsible for most deaths. Glioblastoma, indicated here on the slide in green, is the most common malignant glioma, predominantly manifesting in patients over the age of 50. Unfortunately, prognosis is really dismal. Treatment is typically surgical resection, followed by chemotherapy and radiotherapy. However, relapse invariably occurs as aggressive therapy-resistant cancer. So there's an urgent need for improvements in treatment. Next slide. In recent years, there's been substantial progress in understanding the molecular pathogenesis of gliomas, both in adults and in children, as indicated on the right part of the slide here. So these advances have resulted in improved diagnostic classifications based on the mutational profiles that we can access via our assays. And the improved understanding of the molecular pathogenesis has identified new therapeutic targets which offer the potential for improved outcomes. Next slide. So we've talked about the test directory, and the test directory for neurological cancer is very substantive. And so all of the 7 GLH centres are tasked with delivering multiple tests to improve the need for brain cancer patients. Next slide has got a bit more detail on this. So the bar plot on the left here shows the number of clinical indications, the number of diseases, specific diseases, that are available within the cancer test directory for those different specialties indicated by the different colours. So for neurological tumours, there's 31 distinct tests, and the dot plot shows that for each of those tests, there may be one or more specific tests that can be performed. So typically in each of these 31 clinical indications, there may be 5 or more tests. In some cancer types, there's very many more, 20 or even 30 tests. So on the next slide, we can see out of those 31 indications for neurological cancers, there's actually 374 distinct tests. So it's a complex and expansive necessity to ensure that we access these tests and these target genes appropriately. On the next slide just gives a bit more detail of these 31 indications, and the x-axis shows the number of tests that were mandated to access for each of those specific clinical indications. The colours indicate the class of variant, and there's lots of orange and red indicating copy number changes and structural variants, which are particular classes of variants which whole genome sequencing is highly adept at delivering. Whole genome sequencing is available for each of those, depicted in yellow for each of these tests. And the thing about whole genome sequencing is we're not constrained to the test directory, so we can explore the entire genome. So performing a whole genome sequencing on one of these clinical indications is the same as exploring it on all of them. In the next slide, this just indicates the test directory entry for glioblastoma. So what we see here is 14 different tests. In orange are NGS-based tests, in blue is WGS, in grey are non-NGS-based tests. And those genes depicted in red are genes which are essential, mandated for us to pursue via different technologies. In white are genes which are desirable, so they are genes which may add clinical value. Via panel-based tests, it's unlikely that those genes would be accessible. The focus would be mainly on the essential tests, but via whole genomes, we will capture all of the genes on this list here, and additional genes that might not even be on this test directory thus far. On the next slide, we can see that if we deploy whole genome sequencing, we can simplify the test directory to essentially 2 components. Whole genome sequencing can access all the genes, all the chromosome variants, and different DNA signatures, MSI and TMB. It can access all that variation in this singular assay, other than focal and global methylation profiling that's indicated in grey at the bottom here. So it's a highly insightful test that we can deploy. On the next slide, I can just summarize that. So WGS currently is available for brain cancer patients, and it's the most valuable singular assay that we have in our disposal. It's agnostic and it enables expansive analysis. We extract all gene variations simultaneous with absolute maximum clarity. We can also detect all the chromosomal changes with high resolution and additional readouts such as mutational signatures, MSI, TMB, HRD, and full clarity to germline variation. And contrary to popular belief, a whole genome sequence, the interrogation of 3 billion bases, is not a difficult thing to analyze and report, particularly for brain cancer, where we have a focus on treatment opportunity. It's a really rapid test, semi-automatable, and we really aim to deliver this really quickly to our patients. So I'll just give one clinical vignette that will illustrate the differences between panel-based testing and whole genome sequencing in a particular brain cancer. So this scan is from a 2-year-old who presented with a posterior fossa tumour. The histology favoured an atypical teratoid rhabdoid tumour, or ATRT. On the next slide. The first data that we were able to explore in this case was panel-based data. So we looked at 523 genes, and the genes responsible for this condition were not evident in the data, so there was no variance in genes SMARCA4 and SMARCB1. So we issued a report saying we couldn't support the diagnosis, but we couldn't add additional value other than that. The very next day, the whole genome sequencing appeared, and that's on the next slide. And what we see here is a profile of a relatively quiescent tumour, with the exception of complexity on the right-hand side involving chromosomes 19 to 21. So this makes it suspicious that this complexity underpins the genesis of this particular cancer type here. And indeed, when we explored it in a bit more detail on the next slide, we can see that amidst that complexity, we're able to identify a high-level amplification in a gene called PLAG-L2, which was recently reported in 2022 as a new brain cancer entity. So we're able to secure the diagnosis via a gene which is not even on the test directory, and offer a much more insightful report via access to this test. On the next slide, so we can see how our genomes being adopted generally across different disease types across the GLH network currently. So the good news is since 2021 up to about 2024, the progress is really good. So the number of genes performed is escalating, as we can see. If we look at the next slide, reciprocally, the turnaround times is decreasing. So what we're seeing here is that over time, the brown dots indicate that most genomes are delivered within 2 weeks. If it's a purple dot, they're delivered within 3 weeks. So we're able to access these tests, and they're delivered really reasonably in a reasonable time scale back for us, our analysts, to perform their investigations. In the next slide, we can see that, so does this activity mean that our brain cancer patients are gaining access to this maximally insightful test? So this report, there was a report commissioned by Katharina Kurian and colleagues with working with the Tessagile Foundation, and they approximated that 11,000 patients are diagnosed with a brain cancer each year within the UK. However, on the next slide, we can see that they estimated that on average, less than 5% of adult brain tumour patients were receiving this highly insightful assay. So the next slide provides a bit more resolution on that across the GLH network. So what I'm showing here are the different GLHs across the x-axis here and the number of genomes performed since genomes were adopted in the y-axis here. And we see these spikes indicating inequitable delivery, inequitable access to patients. In the next slide, if we focus on brain tumours specifically, we can see that the first two bars, central and south and the east, are delivering lots of brain cancer genomes, but other centres are delivering far fewer. If we look at genomes within the East generally, it's really quite a success story. So these two slides just illustrate that in more detail. So as well as doing lots of brain cancer genomes illustrated in orange, we also do many paediatric genomes, more than any other GLH, illustrated in purple, and many solid cancer indicated in green, and also sarcoma indicated in pink. And the The really good news about the East is that all of those genomes are really prioritized for analysis, and we don't have any outstanding cases that are not reported. They're all taken to the first available MDT as soon as the data are returned. On the next slide, we can see, so although I indicated it's a hugely valuable test, the reality is that most adult brain cancer patients don't gain access to this test, and there's geographical inequity. Consequently, treatment opportunities may be limited. So to address this, on the next slide, we can see that just a couple of months ago, we convened a workshop and invited clinical colleagues, oncologists, pathologists, clinical scientists, representatives from the charity sector and patient advocates to discuss how we improve the deployment of whole genome sequencing to the benefit of brain cancer patients. And on the next slide, we can see that there was a series of talks, but in addition, there was 3 work streams that were deemed as critical. So we had focus sessions concentrating on those work streams. On the next slide, we can see that for each of those work streams, critical improvements were recognised. And these included, with respect to sample collection, streamlining consent, streamlining tissue processing, potentially introducing RNA later as a liquid medium that can replace fresh frozen, the necessity to fresh frozen tissue, the necessity for dedicated genome coordinators to support this activity. With respect to analysis, it was recognised that local GTABs are useful, particularly to inform diagnosis. Reporting needs to be really rapid and accessible and succinct and therapeutically focused. And long-range sequencing and methylome analysis is something that is likely to play a part in the future, and there'll be talks on that later today. With respect to precision trials, there was a real focus to have a centralized GTAB for precision trial entry, support for local trial deployment, and working with pharma so that new drugs can be adopted into clinical trials as soon as possible. On the next slide, we can see some of the work that's being performed to date with respect to improving outcomes via the adoption of new clinical trials. So Richard Mayer, CR UK surgeon and clinician academic, has been working with the Royal Marsden, with Juanita Lopez, to set up new clinical trials, which are shown on this slide here. The aim is to enable WGS to identify variants which can be discussed in a single national GTAB, focusing on clinical trial deployment. 3 of these clinical trials have already acquired MHRA approval, and these will be opening shortly. And patients can be triaged by this singular GTAB, and so that they can access these trials most effectively. So the consensus statement of our workshop was this, and this is what we would like to see to happen for brain cancer patients. Every UK brain cancer patient should have access to whole genome sequencing and timely molecular analysis to access nationally conducted, molecularly stratified precision medicine clinical trials. So we can aim to try and improve the outcome from these patients. So to summarise, the East runs an effective WGS programme for eligible cancer types. Active engagement to support WGS uptake nationally is a focus for us within our region. We may host a national brain GTAB to triage patients for prospective clinical trial access. And the ultimate aim here is to improve the outcome for brain cancer patients. And in my last slide, just to acknowledge this is a collective endeavour of many people, many scientists, clinicians, and other staff to ensure that this is a deliverable opportunity for patients within our region. Thank you.
Thank you very much, Patrick. For our next session, we get the view from a lived experience, Victoria Smilyk. So Victoria discovered that she carries the BRCA2 gene following genomic testing on the 100K Genomes project, and as she sought a diagnosis for her son's rare condition, Victoria will be in conversation with Professor Julian Barwell, who has been involved in her care at the University Hospitals Leicester.
Thank you so much, Emmanuel, and thank you so much to Victoria for joining us today. Victoria, can you hear me?
Yes, I can.
Fantastic. So Victoria took part in the 100,000 Genome Project, and what we hope to be able to explain today is what it's like for an individual who goes into a project for a particular reason, but it opens up a whole series of other types of questions. And what I want to really sort of drill down on is how she felt going into that and what she was told at the time about the potential results that might come through, and then the support that she got through that process, and then any learning for clinicians and future patients as to how we can reflect on that journey for her. And I really want to thank Victoria because her story is still ongoing, and most patients who tell their story have reached the end of a particular part of that. And so this is a very much an active situation for Victoria. So thanks so much for sharing that with us, Victoria. So can I just start by asking, how did you get involved in the project for you and your family originally?
Well, my son was the reason why we got involved in the project, because he had a range of different needs, and someone recommended that we came to genetics, which is what happened. We were referred to genetics, and then myself and my fiance, got tested in the process, really, to see if we could try and get a diagnosis for what was going on with my son.
And at that time, do you feel on reflection you were aware that you could be found to be a carrier of something completely different?
To be completely honest, I kind of never thought of myself getting a diagnosis. I thought that we may get a diagnosis for my son, but then we may never find out what was going on. And actually, by us taking part, it's realised what could have happened, but actually a lot has happened to prevent me getting what's gone on.
Okay, so how long did it take for that result to come through?
It took 9 years to get.
Wow.
The result.
So that is a long time. And how did you find out when this change in the BRCA2 gene was identified? What was the process?
Well, I was asked to come in to the clinic to have a conversation with one of the consultants, and I spoke to her, and then from there the ball kind of got rolling, really. I was referred to a breast consultant at the Glenfield Hospital in Leicester, and then I was referred to the Leicester General Hospital to have like my ovaries and my tubes removed.
Okay, and did you have any particular problems at that time? Any medical problems at that time that you were referred to talk about surgery and your options?
I had a range of health needs, but kind of never on reflection took into consideration what they may be.
Okay.
I kind of just carried on with what was going on, really, because I've got an autoimmune disease, and everyone kind of put my conditions down to that.
Yeah, because I think you'd had your abdominal problems that were a bit non-specific and hadn't really been sorted out. Is that right?
That's right.
Yeah. So after that, what happened when you went to see the gynecologist?
Well, I saw the gynecologist, and they mentioned about having my ovaries and my tubes and my ovarian cysts removed, and it took a while, but I pushed to have my operation, and then at that point I had my operation, and they told me when I'd just come around in recovery that they'd found a mass on my appendix, And then kind of it went from there, really. And now I'm in the process of being referred to hopefully Basingstoke to see how we move forward.
Okay, so they found a beginning of something which they feel that if left may potentially cause quite significant problems for you, I understand. Is that right?
That's right.
Yeah. And so the you're not sure as of yet if anything further is required, but you were told that if it had been left, it's likely that that would have been an absolutely major problem.
Definitely.
Yeah. So what's happened is you've gone in looking for one issue, and in fact it's found a susceptibility to something else. And then when that's been looked at, it's actually found a third issue,
Right.
which is something that obviously has been picked up almost certainly in the nick of time.
Definitely.
So how has that left you feeling about your own health journey and for the family?
Well, it's been a bit of a roller coaster, really. I kind of expected to find a condition with my son, but actually my journey continues. You have one operation, which then leads to something else. One chapter closes and then a new chapter opens. So in a bit of a spin, my head's in a bit of a spin, but kind of just going with what's being given to me at the moment.
And do you feel there's anything that we can learn from what's happened to you? Because obviously you've had two unexpected findings in the story you've talked about today. Is there anything on reflection during the consent process or when the results were given or since then? Is there anything further you think would have helped?
No, not at all. The genetics team have been absolutely amazing for myself and my son. And no, genetics have been absolutely amazing for me. I wouldn't change anything. The support's been amazing.
Yeah. And with regards to consent, obviously, when you're undergoing these tests, there's all sorts of things which could be found, but they're quite rare. Do you think we need to say more to patients in advance, given what's happened to you, or do you feel we got that balance right originally?
No, I think the balance was right. You go in for one thing, and if you find something else, for me, it was let's get it sorted.
Fantastic. And is there any sort of final message to the healthcare professionals who will be supporting patients through these types of tests in the future? Is there anything that you'd like to say to the healthcare professionals so that they will be able to take that forward when they talk to new patients?
No, I think the consent and the process in which I've gone through, if other hospitals operate the same procedure, then no. It's been amazing for me.
Well, thank you so much. It's a real privilege to talk to you about that, and the very best of luck with the Basingstoke team. Let's hope that nothing further is required, but also best of luck for your son moving forward as well with his clinical issues. Thank you so much.
Thank you. Bye.
Thank you, Victoria, for sharing your experience. I think we are continuing with yourself, Dr. Julian. I guess just a brief intro, which I didn't do earlier. So Julian is a consultant in clinical genetics at the University Hospitals of Leicester, with a particular interest in public engagement, stakeholder group co-creation, and implementation of genomic medicine projects. He helped coin the phrase the Angelina Jolie effect and launched the Cancer Genomics branch of the 100 Genome project on the BBC Breakfast Sofa. and has also co-authored a children's book, What is DNA? Welcome, Julian, again. Over to you, presenting on pharmacogenomics in cancer.
Thank you so much. So at the beginning of the pandemic, I went back to the wards to help. I was all very excited, and it was the first time I've been back for 19 years, and it was all going very well until a young nurse said, "Have you come out of retirement then?" So that was the end of that. But one of the things I noticed was there's a bit of a disconnect on the ward and in oncology outpatients between molecular biology and clinical reality. And so what I wanted to do was to try and help that connection. And so what we're going to talk about today is building on what Anna mentioned earlier, which is the Lynch syndrome app, and how we can use these types of technologies to make sure that people are receiving the right drug at the right dose first time, every time, and really start to deliver on 21st century healthcare. Next slide, please. So the problem we're trying to solve, and this is with two companies that we've set this up, Instant Access Medical and Signogene, is the problem that so many of our medications, actually, when you look at it from the way in which the body will break down and use the drugs, don't actually work, or in fact cause problems. So it may be that the drug that someone's receiving is likely to cause side effects, and Srini talked about that earlier, or actually it may not be the right drug at all, or it may just not work at all, and we'll be going through some of those types of situations. The second issue is that if you get a PDF report, that may not be available when you're in a pharmacy at 5 to 5 on a Friday and there's a long queue. So how do we get to a point where we're giving people the correct drug? Next slide, please. And so the challenge is that on one hand, patients, and we did this with an Ipsos Mori survey with 106 patients from Lynch syndrome UK, is that people are really keen to use healthcare apps, but there are concerns about the safety of these types of apps and whether or not they could be breached, the safety from a governance perspective. But also we know that giving people the same drug and all at the same dose is not a way we can really sustain in the longer term. It's positively Elizabethan, and I don't mean Elizabeth II. It's certainly equality, but it's not equity. And of course, with the rise of patient mobile phones and AI, there's a real opportunity for patients to feedback their experience. And this is part of 4P medicine. If you can predict what is going to go on with a patient in the longer term, then you can personalize and you can prevent healthcare problems, but it only works through participation. And those are the 4Ps of 4P medicine. So this is what we're trying to develop moving forward. Can we have the next slide, please? So the solution is a traffic light system: red, amber, green. So red, drug not safe for the individual, do not prescribe. Amber, safe but maybe need a dose change because of the way the body breaks down the medication. Green, carry on. And we believe that 1 in 6 prescriptions would need to be changed by this approach. But there will be significant issues with regards to clinician and pharmacist and patient training to be able to do this. And we want to build on the work we've done with the Lynch syndrome facing app. The Lynch syndrome app has gone through all the approval process and have been agreed by Lynch syndrome UK, about to release the access code so individuals will be able to download this. And we think this will be better not only for the doctor and the patient, but also to give pharmacies the sense that they are moving into 21st century healthcare. And that will be better for reducing side effects and wasteful prescribing, could reduce insurance costs as well moving forward. A bit like if you have a car app, which helps reduce your premiums because you're driving more safely. Next slide, please. So the product is a multilingual app for Lynch syndrome, which will use Signagene pull through data from pharmacogenetics. An individual will have sent off a saliva sample, and we've developed a training platform and a feedback system. And the idea in time is that you could go into a pharmacy, and this will be a prescribing aid so that you'll be able to check your prescription against your pharmacogenetic profile. But actually, we could use the rare disease app profile whilst you're waiting in A&E, or if you have ischemic heart disease, or for instance, a cancer survivorship app as well, which will link your clinical with your molecular data and have effective signposting and reminders of appointments, and also red flag symptom checkers, for instance. Next slide, please. So this is what it looks like, for instance, for Lynch syndrome. So you have a red, amber, green system here. So across 6 domains. So 3 points for green, 2 for amber, 1 for red. So you get a score out of between 6 and 18. And this is patient added data. So we've got bowel screening, taking aspirin, Helicobacter, talking to relatives, Gini review as appropriate for women, and symptom awareness. And you get a score. You can see there the reminders of appointments as well, and the patients can be signposted to additional information. On the right hand side, you can Trini talked about TPYD, and you can see how you could pull through genetic data, so we could check that this patient is able to take the drug. They've got a green rating. And in 3 or 4 seconds, very quick, you can see how you can check a medication for that individual. Next slide, please. So education is an important part of this. We have a genomic scholar group, which meets monthly, sort of a Dead Poet Society group, where we challenge the very brightest medical students to think about the educational implications of AI in 21st century healthcare and our changing relationship with knowledge. And Jaden Wong is utterly brilliant. He's done a MRC Welcome Summer Scholarship and helped us with some of the educational aspects of this. And you can also see here a rating system. So we want patients who are using the system to feedback their experience with the drug. Does the drug dose need to be changed? Were they happy that they were able to tolerate it? And did the drug work? And you get a 5-to-1 rating. So in time for a new user, they will be able to check how other patients with their same genetic profile to them got on with the drug. So effectively a personalized Amazon rating for your prescription. Next slide, please. So we've developed this training guide so that individuals will understand the role of pharmacogenetics in whether an individual is likely to be able to respond well to a particular medication, being aware that there are other factors, obviously, as well, the other medication that they are taking. There may be ethnicity factors as well, although most of those are related to pharmacogenetics. Next slide, please. And what we have is a sense of urgency, because we're now at the point where for DPYD, you cannot prescribe unless you've actually got this result. But also in America, doctors are starting to get sued. Clopidogrel is a very important example because it's given to reduce the risk of blood clots, but you don't know that the drug isn't working until you have a clot. So it's a preventative drug. It's not that it causes side effects as such. It's just it doesn't work, and there's no biomarker. With a statin, at least you've got a cholesterol, and the same with warfarin with the INR. But the issue, for instance, in Leicester's, there'll be many patients that just won't respond adequately to clopidogrel, and you would never know. And we will look back and think it is utterly incredible that up to half the patients in Leicester City would be given this drug and it may not be working. So we need to get to a point where not only are we offering this test, but we have a dynamic solution so that we can get to a point where we can check that prescription, not purely just relying on a PDF. Certainly doctors will be not thinking so much about whether or not to do it. It's that they will be asked, why didn't you do it in the longer term? And we want a solution that we're ready for when that time is available. And we think that's very soon. Next slide, please. So we have a training guide which has a question. So we've got a training that's linked into the questions linked into the training. So you have to answer them as you go along. And then there's 10 truths and false at the end where you have to get an 8 out of 10. And so hopefully we'll be able to work with Gemma at the GMSA so that we can have this into a format where individuals can get a certificate for pharmacogenetics at the end of a half-an-hour session, so they feel comfortable not only with the medical aspects of this, but also with regards to using these sorts of patient-facing guides, because we think the fundamental principle is that the patient needs to be heavily involved in their clinical care and access to their records. And this is going to be important for driving through these types of digital solutions. Next slide, please. So the reason why it's important now is not purely because of legal implications and the fact that these tests will come onto the test directory. It's because once you are in a position where you can start linking clinical data with molecular data on computers, then there is opportunities to link to healthcare outcomes. and that provides the opportunity for AI learning. So we can start to make each data point count with consent, of course, so we can better understand which patients are going to respond to their medication. Next slide, please. And this is part of a much wider strategy which we have been doing with the Genomic Laboratory Hubs, the 100,000 Genome Project, in partnership with Joe Lowry, our genomic manager, who I've worked with many years, and part of our outreach and clinical support. So we go out into communities to talk to them about building trust and co-creation projects. We've written textbooks, both clinical, but also for children, and we've reached out into the community also through the media. We have another rare disease group, P10 UK and Ireland, who have agreed in principle to use the Lynch syndrome app. We're in discussion with the National Hereditary Breast Cancer Helpline about developing a rare disease app for BRCA and PALB2 gene carriers. And as I said, we're underpinned by patient surveys with Ipsos Mori, and certainly feel that we're in a good position to drive through these types of 21st century healthcare solutions for individuals, not only with rare disease, but also on the high street. And we've just been awarded a program grant to hopefully be able to look at decision aids in other sections as well. So thank you very much for the opportunity today, and I will be staying on for the question and answer session later on. Thank you very much, Emmanuel, and for everyone involved. Thank you, Julian. Our next session, Supporting Healthcare Professionals, Education and Training, will be delivered by Dr. Gemma Chandratillake, who is our Education and Training Lead at East GMSA. Gemma trained as a molecular geneticist and a genetic counselor. She has a long-standing interest in rare genetic diseases. She is chair-elect of the British Society of Genetic Medicine,
and coordinates the master's course in genomic medicine at the University of Cambridge. Welcome, Gemma.
Hi, thank you, Emmanuel. All right, I'm ready, Ian. Okay, so today we're going to talk a little bit about education and training of the healthcare workforce with respect to cancer genomics. So if we're thinking about cancer and the workforce that delivers cancer care, it's a complicated workforce. So, lots of different healthcare professionals are involved in delivering care for patients with cancer, and therefore we need to reach a lot of different professionals about genomics and explain and help them to integrate genomics into their care. So I'm going to talk about some of the particular interventions that we've been doing at national level and at regional level today. So can you hit that? Thank you. And the next one. So we're going to be thinking about the staff that work in genomic services. So those scientists that are working in our laboratories who are experiencing an increased demand for existing tests, as well as you've heard today, as a demand for new tests, as our understanding is developing. Thank you. And we're also going to be thinking about clinicians who are dealing with the ordering and the interpreting and the communicating and counselling and acting on these test results. So I'm just pulling that from HE's Strategic Cancer Workforce, which has genomics threaded through it. Okay, so we'll start off by thinking about the laboratory staff themselves. Okay, next slide. So back in 2020, as we were bringing the genomic medicine service, getting it up and running in our region, we did a piece of work looking at our workforce in the laboratories across Cambridge, Leicester and Nottingham. So Cambridge is blue, Nottingham is green and Leicester is purple. And we were just looking at all the staff in all laboratories that we were bringing together as the Genomic Laboratory Hub. And we found that there was a relatively low number of staff that were actually working in the cancer testing aspect of the laboratories. Next slide. We also did a huge piece of work assessing all of the staff's knowledge. So what do they know about genomics? This is the laboratory staff that are delivering the testing. And they did a big piece of self-assessment, and so that we would understand how many people in our region, in the laboratories that we're delivering the service with, how many people kind of knew about cancer genomics, because it was a new service really that was developed. And you can see from here, next click please, that we have quite a low number of laboratory staff in Cambridge, Leicester and Nottingham, genomics labs that actually had good knowledge of cancer genomics, that was secure in their knowledge. Next click. And that's really because there'd been a historical lack of investment in staff up-to-dateness. So staff hadn't really been supported in learning about, there hadn't been a lot of horizon scanning and supporting staff to learn about new technologies and new areas of implementation. Next slide. So we knew we had a lot of work to do because we have a small number of staff with this expertise. And these are the staff that are delivering the NHS service, but they're also the staff that are adopting the new technologies into the service. And they're also the staff that are training staff. So there's a lot of pressure on the small number of individuals, and we need there's a balance that's required between these different aspects, but they can come into tension with each other because we had low staff numbers. So we really had to kind of bring a cultural shift with respect to the workforce, where we have adopted an always training mindset with everybody in all the laboratories. We also put in a dedicated training team to support the scientists to train as efficiently as possible. And then we've also done a lot of investment in staff development, so funded courses, conferences, formal education and professional registration, really to support staff to be up to date and to support them in their career progression. Next slide, please. At a national level, this is a nationally recognised issue, actually. And so NHS England developed a national cancer genomics scientist training programme that was first commissioned in 2018. It's a 3-year programme, so it graduated the first cohort in 2021. And they are currently developing a national genomic training academy to support training of the laboratory workforce, because as you probably can see from earlier talks today, a lot of new technologies are coming online, and this is an expanding area of work. Click. At the regional level, East Genomics, what we have done is we have cross-trained some of our senior scientists who had been knowledgeable in rare disease genomics. We've cross-trained them in cancer genomics. We've also harmonised all of our training across the region. So all of our staff in Cambridge, Leicester and Nottingham, we co-train them all, which enables us to do it very efficiently and use the expertise across all of those laboratories that we have. We've got a dedicated training team within the laboratories to manage the training and to expand the training capacity. And we've also implemented e-learning program platforms and also training resource platforms across the regional laboratories. Next slide. Okay, so that's the laboratories. Now we're going to think about outside the labs. So if we think about the NHS workforce, we are the largest employer in Europe. We're the 5th largest employer in the world, but there are actually only a small number of people in the system that have a real specialism of knowledge around genomics. So we've got a couple of hundred clinical geneticists and a few hundred genetic counsellors. So we know that genomic services will have to be delivered to patients by non-genetic specialists. And this concept is called mainstreaming. Next slide. So when we're thinking about mainstreaming, we recognise that not all healthcare professionals need to become genomic experts, but they do need to know when a genomic test is relevant for their patients, and they need to know how to request that test and to have conversations with patients about the results. Okay, next slide. So the challenge here is that we knew from surveys that we had done across our region in 2016 and 2021 that in the mainstream workforce, there was a real lack of awareness of genomics. There was a lack of training, and we've seen that kind of echoed today when a lot of people on this call said that they haven't actually had any genomics training. There was a lack of awareness of training opportunities, so people didn't even know what was available to them. And there was also a perception that genomics was irrelevant to their role. So we knew we had lots of work to do. So here's some of the things that have been going on at national and regional level. So at national level, we have the National Genomics Education Programme. Next slide. And they have really taken a multidimensional approach to all of this. So they have done a lot of workforce planning work, workforce development work. They've been funding a lot of training. They've been developing a lot of resources and also engaging with a lot of stakeholders to sort of raise awareness and get genomics into various clinician curricula, etc. Next slide. And this is the kind of work they've been doing. So at the top of the triangle there, we've got formal training. So this is the establishment of the genomic training academies and the funding of scientists to train. And then we go down to the next level with the formal education. So they have commissioned master's programs around the country. These are modular master's programs, and this enables healthcare professionals to take funded education. They can do a master's degree or a postgraduate certificate or diploma in genomic medicine, or they can just take individual modules, and I'll mention more about that in a minute. So that is open, available to NHS staff at the moment. They've made lots of online courses, so people can take up those opportunities. Lots of resources, so fact sheets, films, videos, etc., which helps individual healthcare professionals to learn, but also helps people like myself to train. So I don't have to make all of them from scratch. And they do a lot of work on social media too. Next slide. So at the universities, they have funded genomic medicine master's programs. In our region, in the East Genomics region, we have one university that has been commissioned to run this program, which is the University of Cambridge. I'm involved in that program. So these are the modules at master's level. So these are 1 week long modules. If you are a healthcare professional in our region or actually outside our region, it doesn't matter. You can apply to the university or to other universities around the country, and there is funding for NHS staff to take these modules. So for cancer workforce, these are the modules that I would recommend. Obviously, there's a cancer genomics module. You can also take an introduction module just to get your kind of molecular biology up to scratch. And then there's pharmacogenetics, epigenetics, and counselling skills. So these would all be useful modules for anyone who's working in cancer care. Next slide. One of the other big initiatives that has been done centrally is the creation of Genotes, which is a just-in-time educational resource for healthcare professionals, and it's designed to be used in a clinical setting. So Genotes is being rolled out for every medical specialism, but already live we have oncology, primary care, and pharmacogenomics. So in terms of cancer, these resources are available. So if you're not familiar with Genotes, just Google Genotes and have a little poke around in the oncology section of Genotes, and you'll find that there's lots and lots of guidance around how to manage your patients. So we've been thinking about also how to incorporate genomics into regional medical training. Next slide. So we've been working with the deaneries in the East of England and the East Midlands. If we think about how a doctor is trained, they go to medical school and then they come into the deaneries for their follow-up training. So we have the foundation doctor training, then we have the specialty training programs, and then those are fully qualified doctors now, but they also, but our consultants obviously need training in genomics as well because they wouldn't have received that earlier. So we've been working all through this pathway. Next slide. In the foundation level, we have been delivering half-day educational events since 2018. We run about 4 of these across the region every year, and the feedback has been very consistently good. This is for all foundation doctors, so not particularly cancer, but we catch everybody who's going into oncology as we catch everybody who's going into primary care as well. So the advantage of this approach is that it's a pan-specialty approach. We've also been working with the deaneries looking at the training programs. So we did a survey of the training program directors in 2021, asking them whether their regional training program actually incorporated genomics. And the thing to look at here is this pale blue, very thin slice, which is those who answered yes to that question. Everybody else either answered no, or not yet, or we're working on it. So there's still a lot of work to be done there. Next slide. And thinking about our responses from those involved in the training of cancer doctors. So it was good to see that histopathology was already one of the yeses. So they had incorporated genomics content into the training program, but sort of medical and clinical oncology, they were very aware that this needed to happen and they were working on it. So they were certainly ahead. I think in cancer, people are really do recognize the need for genomics in their care of their patients. So I would say that they, as a specialism, they're kind of ahead of the game in incorporating genomics into their training. Okay, next slide. Some of the other things that we've been doing at the regional level is we have developed some e-learnings. It just enables us to train lots of clinicians at scale. So we have a couple that are relevant to cancer. One is our patient choice, discussing whole genome sequencing with your patients. So you've heard about whole genome sequencing earlier from Patrick. So if you want to be ordering this test for your patients, it's important that you train in how to have a good conversation with patients about that test. So this is really targeted at oncologists and nurses. So you please do take this e-learning to cover all the things that you should be talking about with patients. And then we also have one on genetic testing for hereditary breast cancer. So this is really targeted towards breast cancer teams. So these e-learnings are both available for you to take if they would be useful. We have also been sort of exploring other options of delivering training. So at the moment, Genotes is a web-based browser offering. It is planned that they will expand Genotes to become an app, but at the moment it is on the browser. So we have been working with QGenome to have an app-based educational offering as well. So you can download QGenome onto your phone, and there is education for cancer genomics and guidance for primary care around cancer as well through that mechanism. So that's all linked to the National Genomic Medicine Service recommendations. Okay, so that was a bit of a whistle-stop tour of all the kind of scoping work that we've been doing and the resources that are available. And if any of your teams need any training, please do reach out to us at the GMSA, and we'd be happy to deliver it for you and point and do some signposting as to other resources that are available. Thanks.
Thank you very much, Gemma. On to our next session. Sorry, we're running a few minutes behind, but hopefully we'll catch up as we go along. is the Streamlining Pathways to Precision Medicine in Lymphoma talk. This is part of the Networks of Excellence project, delivered by Dr. Livia Raso-Barnett, a consultant hematopathologist in the Cambridge HODS service, and the clinical co-lead for the HemOnc at East GLH, with a particular focus on lymphoma. Welcome, Livia.
Thank you very much, Emmanuel. So we change pace a little bit. I'm not a scientist and not a clinician, as you heard. I am a diagnostician, and I'll promise you we're going to get to the network of excellence, but I think for you to truly understand why this is important, it will be important to understand a bit of a background of why lymphoma is where it is. Next slide, please. So when we're looking at genomic testing, we usually are focusing roughly 3 large aspects of genomic testing: targets and testing of diagnostic importance, that of prognostic importance, and targeted therapy importance. The reason why lymphoma is unique is because even though we're talking about 2 cells only, the B cells and the T cells, according to the current iteration of the WHO classification, there are now 90 different recognized individual lymphoma types. And therefore, you can imagine if you want to get to the correct diagnosis that will be followed by the correct treatment for the patient, you have to do quite a bit of complex testing and working up. All of the part of the reason why lymphoma is also very special, because that molecular aspects of diagnostics has been incredibly fast growing over the last 20 years. And you'll see that's exactly why it's also very difficult to implement it nowadays as well. So as I said, we do an awful lot of working in the histopathology lab, use of a lot of tissue. And even if you rationalize what molecular testing you do just for clinical purposes, just the ones that are absolutely necessary, you still need to do at the diagnostic stage molecular testing at least a third of the cases. And another challenge is that even though it's slightly variable across the country, most hospitals will only receive a minute percentage of excisions or nodes. Most of our samples are tiny core biopsies. So if you can imagine that even a tiny biopsy, you do an awful lot of histochemical works on that will need to do molecular testing on as well. It can be really, really challenging to get to the correct diagnosis. Further complicated is the fact that we're not going to get an excision in lymphoma. The patient will be treated in that tiny biopsy and the information we can get out of it. And of course, if you talk to any specialist in a clinical setting, they will tell you how life-threatening their specialist diseases are, because B cells and T cells and lymphoma can occur anywhere in the body. All of that applies to lymphoma as well. Even more, we have some very fast-growing tumors, which if they break down can be immediately life-threatening to tumor lysis syndrome, can be putting the patients into IT, they can be incredibly sick. So we do a lot of complex testing on tiny pieces of tissue, and we get to the correct diagnosis as soon as possible. Next slide, please. And just because I'm a visual person, I put that all in a slide for you. So unlike other cancer types, we don't just need to get to a yes or no cancer diagnosis. We need to get to if it's cancer, what type of cancer, because before we can get treatment. And oftentimes we will have done a lot of testing in the immunohistochemistry lab and molecular lab and still don't know the answer, have to go back to the beginning and take a biopsy, which can be quite time consuming, as you can imagine. Next one, please. And this is exactly why there was a first round of centralization for lymphoma and hematological malignancies about 15 years ago, recognizing this multimodality, tight turnaround time workup is required. And hematological malignancy diagnostic services were set up across the country. That's what HODS pods is here in Cambridge. Next slide. So what this means in practice is that our center here in Cambridge receives samples from 10 different submitting laboratories and hospitals. Next slide, please. We already are networked. And of course, now to ensure timely and equitable access to genomic testing to everybody, we now have the GLHs and associated GMSs set up. All the testing indications and the set of targets are centrally set, so everyone can have the same level of access to everything. And this is then contained within the test directory. Next. For lymphoma patients, from a practical purpose, what it means is we have 3 HMDSs, i.e. we have 3 already networks which network into the GLH. So lymphoma patients, you receive samples of it in the GLH, come from a network and multiple steps to get to the GLH. Next step. So as you can imagine, this can result in significant differences in diagnoses for patients, depending on where the biopsy was taken. This can be as much as 7 to 10 days. Next slide, please. And of course, not all GLHs are the same. Not everything is networked the same way, and not HMDSs are set up the same way. And these are all variable. And if you add up all the variabilities with varying workload-related pressures across different laboratories, you can imagine that the pressure will mount in the lymphoma world. Also, a further thing to consider is that unlike our counterparts in hematological malignancies, the myeloid disorders, where the samples are submitted straight from clinicians to the genomic laboratory, and unlike our most tissue type related specialties in solid cancer, where the tissue is submitted from pathology laboratories directly to the GLH, for lymphoma patients, they go through a network center laboratory reporting, then onto the GLH. So we have one extra step in the pathway, and all the samples they receive in a genomic laboratory have been through 2 to 3 departments prior to that. Next. And this is partially what contributed to a limited take-up of molecular testing in lymphoma. Not only that, but because the test directory is not particularly tailored to providing help with diagnostics. And quite often we still go to the multidisciplinary meetings for deciding about cancer treatment, having a differential diagnosis, and we just choose the most likely, the more likely diagnosis rather than truly knowing what it is, which then means that if the patients failed the first line of therapy, we do not know if they failed the first line of therapy because they failed it, there's an underlying molecular alteration, another reason they failed, or whether they failed the incorrect line of therapy, which was meant for the differential diagnosis we did not choose at the beginning. Next slide, please. And of course, whole genome sequencing is also available to lymphoma patients, but if you can imagine, if you're a network patient, it will be really, really difficult to organize the right sample to get to the right place. Next slide. You can do a number of things to make sure that you get to the most correct diagnosis, and we've done all of it. We try to strategize, we try to minimize what testing we do as to not to put pressure on the laboratories. We are using equivalent testing wherever possible, but it's still challenging. And of course, knowing that the test directory could be more useful for lymphoma patients, we have submitted change requests to the test directory as well. Next slide. But as you can imagine, if the NHS and Genomics England are paying for a test to be rolled out everywhere in the country, you have to provide sufficient clinical evidence for that to be useful. Next slide. And therefore, if you have tiny bits of tissue that you exhaust, sorry, previous one, if you have tiny bits of tissue that you exhaust for standard of care testing, including molecular testing that's not quite correct, then you're not going to have any tissue left to provide clinical evidence for any additional testing to be useful, and your target's not going to make it on the test directory. Next slide. That's why we were very, very excited about the Network of Excellence opportunity, and working via academic partners and our partners up in Leeds, they've put together a project for lymphoma, which would hopefully get to a better outcome for lymphoma patients. Next slide. And so working along 4 basic lines, we're hoping to address different aspects of lymphoma diagnostics. We're trying to optimize where the tissue travels and how it's handled, so it gets before it gets to the laboratory and while it is in the laboratory, and also come up with tools that are better and more useful when the sample arrives in the genomic laboratory as well, and assess other ways as well that this genomic testing could be used, whether they could replace 1 or 2 other modalities within the laboratory, or whether the testing could be moved out to a different, not identical, but similar methodology with fewer involved departments. And you see in a minute what I mean by that. Next slide. And within this package, we have 4 parallel work streams. One is focused around mutation and expression profiling to get to the correct diagnosis and the most reliable prognostic and targeted therapy information for the patients, the gene expression profiling being done in leads. We also have a work package, a work stream around ctDNA, which you will have heard a lot about in previous presentations, to see its utility in this cancer type with very little diagnostic material available, and also assess how WGS could be built in the process of diagnostic and prognostication, whether that could replace or run in parallel with other aspects of diagnostics. Next. And we have a great local team, including diagnosticians from HODS, scientists from the GLH, as well as academic, clinical and research partners as well. And we're working with one of the biggest HMDS leads on this project. Next. And the first and biggest part of this project, to have a lymphoma NGS sequencing panel that could be as useful as possible. Because knowledge about lymphoma extends at such a rate, it will be very difficult to keep updating everything and re-evaluating on a test directory. There's no custom panel out there that would be useful for this purpose. And therefore, we have come up with an NGS panel design that will include 3 categories of genes. Ones that are already in a test directory, ones that we already know to be of importance, and a third category that we know to be of emerging importance to assess whether or not they would have a place on the test directory. And therefore, we do not need to maintain or keep any tissue for further research testing. This could be done at one setting, and further down the line, a panel can be streamlined and cut down if something is found to be not to be of importance. And of course, this panel's got to work for our tiny, go back please. This panel's got to work for our tiny poor quality samples. And we're hoping as the sample quality improves, we can improve it, potentially replacing other testing modalities and handling within the GLH as well as outside of the GLH. Next slide, please. Because in all honesty, there's not an awful lot of additional targets from a diagnostic perspective that could be there that would be useful so that the patients get the correct treatment from day one. And this could be the difference between watch and wait, local radiotherapy versus systemic chemotherapy. So it is very significant from a patient's perspective. Next one. And again, at the moment, we have tiny bits of samples, which have poor quality because they're fixed and embedded into wax. And as I talked you through before, they come from multiple departments and multiple steps, and everyone's meeting their own turnaround times. But if we could find a way where different tissue types could bypass some of these additional departments, that could be incredibly useful. So if you have fresh tissue, that could be sent direct to the diagnostic center, the hematological diagnostic center. Tissue sending off to the GLH may not even be necessary because there's complementary testing that can be performed within the diagnostic laboratory, which would allow us not to test in the genomic laboratory. Next slide. And of course, this potential pathway may even allow for similar turnaround times for patients regardless of where they are geographically. Next slide. And to make you a little bit more involved and understand a bit more what I'm talking about, I'm going to tell you about this 71-year-old male who had a large mass in the retroperitoneum biopsied. Retroperitoneal lesions are difficult to biopsy and requires CT guidance. And tissue that we get out is even smaller than a cord biopsy, these tiny bits. You can see most of the red in the background is blood, and those tiny blue bits are the tissue we're looking at, that's minus. But thankfully, this patient had 2 samples taken, one informal and sent to the histopathology department, and one fresh sent to the diagnostic laboratory. Next slide, please. And this turned out to be high-grade B-cell lymphoma, which will require FISH testing so that the patient can get the appropriate high-grade treatment. And this patient only had formal fixed sample that would have meant a 21-day to diagnosis and treatment commencement for this patient. However, because we have flow cytometry, we could get to high-grade lymphoma diagnosis within 16 hours as opposed to 7 days, and we could get a FISH done by the time I got the immunosections out on my case, I already had a final diagnosis with molecular classification. Next slide. And therefore, at the end of this project, we hope that we could come up with a number of different pathways and handling guidance that could be applicable regardless of what local challenges or local pathways are within the networks that we're dealing with. I mean, hoping that by the time the samples get to the genomic laboratory, there will be tools that are helpful in providing the appropriate information, genomic information, to inform first-line treatment rather than just failed treatments as well. We're hoping to explore so different service delivery models where testing modalities can be optimized within the laboratory as well as network diagnostic settings as well. In the end of the 2-year project, we are expected to inform the National Test Directory about what are the target and testing indications that are useful or not useful that are currently in the test directory. Thank you very much.
Thank you, Livia. Really great presentation. The next section, we are focusing on some case studies. So we've got 3 case studies, and I'll introduce the 3 case studies and the speakers now, just so that to allow for easier flow. So the first case study is targeting treatments, so pharmacogenomics. This will be delivered by Aris Saoulidis, who is our clinical pharmacy lead, and also a guest lecturer and researcher in genomic medicine at the University of Cambridge. He's currently UK Clinical Pharmacy Association Genomics Committee Education Lead, raising profile of genomics in pharmacy. The next case study, we'll hear from 2 colleagues. So the case study is next generation sequencing, Nanopore. This session is led by Dr. Simon Paine, a consultant nephrologist at Nottingham University and honorary associated professor at the University of Nottingham. He has particular interest in paediatric tumors, teaching, and the application of long-read technology for rapid diagnosis of brain spinal tumors in adults and children. Joining him in that session is Prof Matt Luce, who is a professor of developmental and computational biology in the Faculty of Medicine and Health Sciences at the University of Nottingham. The last session in terms of the case studies is benefits for children with suspected cancer from routine whole genome sequencing, WGS, and this will be presented by Prof Sam Behjati, an academic pediatric oncologist who combines clinical work with research on childhood cancer. Welcome, colleagues. I'll hand over to Aris in the first instance.
Yeah, so hello everyone, my name is Aris Saoulidis. I'm the clinical pharmacy lead for genomics at the East GMSA, and I am job sharing in this role with Paul Selby, who's also a consultant pharmacist. And if you start the first slide, please, Ian. So I would like to talk to you today about how we are striving to optimize the use of medicines through genomic medicine, and just providing a couple of points of what some examples are in practice, and just really going through some of the process of what we are doing in the East GMSA to support the NHS in the uptake of genomic medicine. So specifically around medicines, which is my focus as a pharmacist, is there's a lot of different aspects of where it can be used. So one of the most high topics at the moment is gene therapies, where cutting-edge technology can be used to solve genetic diseases, to treat disease at the root cause of the problem. There's also more targeted treatments, which are prominent in cancer. So we can offer precision medicines that target the molecular cause of a cancer, for example. And then you have pharmacogenomics, which is when we identify variation in the genetic profile of a patient that can be used to guide those treatment decisions, so choosing the best medicine for them and also optimizing a dose for that patient that can predict the drug response for both efficacy and safety aspects. We can also use genomics to repurpose medicines, so identifying the root cause of a molecular disease, for example, in genetics, and finding medicines that are not normally used in that condition, and finding the evidence that is evidence-based to use that medicine in such a condition. And then outside cancer, there's also some drug resistance prediction opportunities for antibiotics, for example. And also in some cases, you can use genetics to predict which cancers are going to be resistant to specific medicines and account for that in your treatment protocol. And there's really a lot of things that you have to consider when you're implementing genomics in medicines optimization. So this is one way to say this, which is how you're going to develop your workforce to understand the implications of genomics around the clinical area. Horizon scanning, which means you have to look into the near future to see what's going to come available, to make sure that your systems are future-proofed to account for that. And test evaluation, where you have to make sure that a test is evaluated properly before it's implemented, and how that's going to affect the economic impact of the health system, how you can account for future research and innovation, how you integrate it into clinical pathways and your workflows on a day-to-day basis, where the key is to make sure that there's equity of access in a routine standard of care for all patients. So equal opportunities for all patients to be offered the genomic test when they're eligible for it. You also have to think about the digital infrastructure interoperability of those tests. So making sure that the digital basis of your health system ensures that your patient's genetic information is available at every point of the health service. And also raising awareness. If clinicians don't know that the test is available, or even patients, then they might not access it as much as possible. Next slide, please. And in a similar vein, this is expanded to a few more things like that you have to consider, that we break it down to including ethical, legal, and social implications on the top right, and patient and public involvement, which is my favorite. Next slide, please. So yeah, so as I said, I'll be talking about a couple of high-profile examples that we have in the East, and then a few projects that we are supporting at the national level. Next slide, please. So one of the cancer-related pharmacogenomic tests that are prominent in the NHS Genomic Medicine Service is testing for TPMT and NAT-T15. So in acute lymphoblastic leukemia, there are two drugs used often called mercaptopurine and thioguanine. And these are removed from the body using key enzymes. Our body removes them with the key enzymes TPMT and NAT-T15. These are prone to variation, so different patients might have a different level of those enzymes. So in some patients, they can experience toxicity because of accumulation from those drugs. And we offer this test in the Genomic Medicine Service as of last year. And patients who have acute lymphoblastic leukemia can be offered this and have the dose optimized for the right medicine at the right time. And yeah, next slide, please. But one of the most prominent pharmacogenomic tests in cancer at the moment is DPYD testing for fluoropyrimidine drugs. So these are capecitabine and fluorouracil chemotherapy. They're used in multiple cancers across the NHS, including colorectal, breast, esophageal, hepatic, and head and neck cancers. And there's about 40,000 patients a year that receive these drugs at least once. And actually about up to 40% of those patients will have some level of toxic side effects, which can be expected. But unfortunately, about 1% of them can have such severe toxicity that can be fatal. But about 1 in 3 of those toxicities can be predicted by having a genetic test before they start the treatment. And then we can account for that and optimize the dose for them and prescribe either an alternative medicine in those patients who don't have enough DPYD enzyme to remove those drugs, or reduce the dose enough to make sure that they don't reach those toxic levels. So in the East at the moment, we have managed successfully through the work of a lot of people to make it a standard of care. And now it's routine that every patient who is due to have this medicine has this test, and it's about 4,000 patients per year in the East. And nationally, the figures are that about 7% of patients who have this test are found to have an actionable result, meaning that they require a different dose or a different medicine to prevent some sort of toxic side effect. And this was a lot of work, because it was a transformation project in the East and in a few other genomic medicine service alliances. And now this is estimated to potentially avoid the deaths of about 40 patients per year in the country. And I want to talk about a little bit more about this DPYD, because we did a lot of work that helped us understand how we can manage the implementation of future pharmacogenomic tests. So we use this as a template, if you like, to understand what are the key points that we need to address when we implement such tests at that scale. So this is an example of the pathway analysis that we did at the time. So we talked to a lot of patients, talked to a lot of clinicians, users, and the labs, and identified all the little bottlenecks on the pathway and the different steps of the process, breaking it down by patient experience, how the blood sample and genetic test actually takes place, how the clinical prescribing and the screening of those drugs takes place, and in the end, how you follow up on those data to inform your future practice and make sure that what you're doing is actually the best thing you should be doing. And you can see it's a fairly complicated chart there, but essentially we try to find the most common steps that every trust in the East takes when they're involved with this test. And there's a link to a poster if you want to read more about this there. Next slide, please. And this led to understanding the different steps that a clinical professional takes when they are involved in the DPYD testing pathway. So from identifying when a patient is eligible for that test, when they need to counsel a patient for this test and what this test will mean for the treatment, how to request this test, how to interpret it and understand the results from it, how to talk to the patient about the results, then how the pharmacist will assess this result and amend a chemotherapy prescription together with the oncology specialist and the nurses, and how we can use the patient's whole picture of biochemical results, how to talk to the patient, and how to finally administer this medicine. And finally, assess whether toxicity was prevented, or if they had the toxicity, how severe was it? And in every step of that pathway, we identified nationally as a group all the different skills that a healthcare professional would need to have in order to make sure that this test is delivered as a standard of care across the whole country. And this is called the Clinical Pathway Initiative, and it's actually due for publication as we speak in the next couple of weeks. We also did a lot of work with talking to patient groups in the East and also nationally to understand what the patients actually want to know. So we asked patients who had cancer and patients who had the DPYD test to understand their experience of this pathway. How do they feel that the information was communicated to them, and whether there's anything that we could have improved on. And this led to this poster that's on the slide here. And finally, to a patient information leaflet template that was published on the Macmillan Cancer Support pages that we can now offer to patients. And this helps them to understand more about what DPYD is and how this can help their care and prevent potential toxicity in 7% of patients. Next slide, please. And in the second year of this project, we understood that a lot of the bottlenecks in the system were informatics related. So as I said earlier, you have to make sure that your digital infrastructure is up to speed and as seamless and optimized as possible. And it turns out that a lot of the delays in the system are because currently in NHS, we tend to send PDF reports as results to doctors, and this introduces extra delays at every step of the process. And we try to map this process, how this looks like from a digital point of view. And this is the simplified version of it. But essentially, we mapped how this looks like currently, and this is how we want it to look like in the future, where every system in the NHS is connected and all the software and programs talk to each other, so that the genetic test request can be done seamlessly, so that we don't waste doctors' time and pharmacists' time to request the test. And when the test result is processed, the result can be sent back to the prescribing software as fast as possible. And it's also really important that the test result follows the patient along the pathway. So when the patient goes to the GP or to another hospital, the GP or the other hospital can also find that test, access it, see the result, and see when it was taken, so they can inform other drugs they're prescribing. And this template was recommended to national stakeholders like NHS England, cancer stakeholders, and the NHS England Genomic Medicine Prescribing Digital and Informatics Standards Groups, a bit of a mouthful, which we're continuing to support. And nationally, as I said, the DPYD project was used as a template to inform how the next pharmacogenomic tests will be implemented. Yeah, so another project that the East is closely involved in is around the deliverables of equitable opportunity to access genomic testing that will later inform the use of precision medicines. So we have to make sure that every patient has equal opportunities in the patient pathway or when they see the doctor to be offered the appropriate genetic test that is eligible for their condition. And then this will hopefully unlock the best treatments possible, the precision medicines for that patient. And this is another template, a project that we're using for that is in non-small cell lung cancer, where there's about 12 different drugs and multiple genes involved in the test panel. And currently we're trying to understand the factors that help clinicians order the genes, the gene tests that they should order, and to make sure that all the clinicians in the country understand what medicines are available for those patients with specific results. And again, as a pharmacist, we're focusing on the medicines. So trying to define the practice gaps where medicines are potentially not prescribed as they could be in the best case scenario. Next slide, please. So there's a lot of different steps, for example, that where there's points of failure, as we call it, or where specifically patients might be not, there might be from the full sample of the patients, patients might not be eligible for that test. So we try to map this, and this is an example of an American research paper that showed when you start from 1,000 patients, about 356 in the end are treated with precision medicine that they potentially should be accessing. So we're trying to replicate this in the NHS using the data sources available to us and trying to understand the gaps in the patient pathway to make sure that we're giving our patients the best care possible. Next slide, please. And this is an example of other work going on in NHS in a similar vein. So recently there was the National Optimum and Genomic Molecular Pathway published for non-small cell lung cancer, and this suggests to cancer services what is the best way to conduct your genetic testing. And on top of that, we are working with our cancer colleagues across the country in the East Patch to understand what are the best questions to ask to understand those gaps I mentioned earlier. And this is a work in progress, essentially. Next slide, please. And I just wanted to close off by mentioning the NHS Genomics Network of Excellence for Pharmacogenomics and Medicines Optimization, who are developing several work packages to understand the best way to implement pharmacogenomic tests like DPYD, TPMT, and NAT15 that I mentioned earlier. At the moment, they are focusing in primary care, in general practice across England, but all the learnings that we will get from these will be, again, extrapolated on cancer pharmacogenomic tests and even cancer precision medicine tests. And there's a lot of work packages going on, as you can see. One of them is how to best implement it, how to make sure that a solution is agnostic, as they call it, that applies to several different prescribing systems, how to make sure that patients are empowered and they know what's going on about those tests, they know what this means to have this test, and they can consent to those tests appropriately. It's also very important to make sure that tests are cost effective, and we're making the best use of the NHS money, so there's a health economics package. And number 5 is making sure that the data that we produce and how we can follow up on those, the implementation of the test is optimized, but also taking into context that it's a real world setting, so you can't have the best case scenario when it comes to data capturing after you have implemented it in the real world. And I think these are the highlights for pharmacogenomics in cancer. And if anyone wants to get more involved or wants to find out more information about specific projects, please get in touch. These are our emails, and that opens up an email to send to me. Thank you.
Thank you very much, Aris. Hand over to Dr. Simon Paine and Prof Matt Luce.
Good afternoon. I'm not sure if my camera is going to work. So my internet here is pretty ropey. Let's see. So Matt and I are going to talk to you about a project on next generation sequencing and using Nanopore based sequencing to diagnose brain tumors. If I could have the next slide, please. And the one after that. So thank you. What's the challenge? Well, there are about 11,000 new brain tumors a year in the UK, and they can be really challenging to diagnose. Patrick spoke brilliantly on the whole genome aspect, and we're talking much more at the sort of diagnostic emphasis of the molecular changes that we need to assess. And brain tumors are particularly difficult on that front. And if I could have the next slide, please. And there are a number of reasons why they're so challenging and why molecular analyses are absolutely key to doing what we used to do with a microscope. So tumors that look the same can behave completely differently. I won't go through these in any detail just because of time, but we can have tumors that molecularly are very distinct, but yet down the microscope look exactly the same. And then the reverse is true. So tumors that look very different, something that looks quite low grade down the microscope can actually, from a molecular perspective, have high grade features. And that's now embedded in our current WHO classification. And then finally, as Patrick showed that Plagel-2 case earlier, there are many tumor entities for which there are no distinctive morphological features. So down the microscope, you can't diagnose them. And this is an example of that. This is a young child, I think about the age of 6 or 7, when this tumor was diagnosed. And you can see it's got a range of features to it. And I diagnosed this several years ago before we had available what's below, which is the methylation array. And the principle that we now use to diagnose many tumors is that the epigenetic changes, the pattern of methylation across the genome, is distinctive for tumor types. And this is all based on fantastic work done in Heidelberg by Felix Salm, David Kapper and co, who built a library of known knowns, and then you compare your methylation fingerprint to that library for your unknown case, and it gives you a diagnosis. And so I called this tumor something called a malignant ectomesencymoma several years ago before we had that, and we now know that this is a PATZ1 fused neuroepithelial tumor. And on the basis of that, the patient can then be treated, family counseled, etc. Next slide, please. So molecular chain testing, particularly methylation array, are absolutely key to diagnosis. And we don't have access to them in the East. We have to send our cases to London. And the pattern, the model of testing has not been designed. It's the way it's sort of organically grown to meet need based on a few centres that are able to offer it. And so if you flick to the next slide, you'll see that we, like many others, have to send all our cases to London, to Queen Square for methylation array. And the actual array itself takes 14 days or so, but in between, or on either side of that process rather, are other delays. So the sample transport, the London lab will only accept our cases once we've issued our report. So we've got to get all the immunohistochemistry, get the case reported, then sent down to London, received in London, etc. The array itself, and then the report comes back to us. And our experience is that that takes several weeks for that to happen. And of course, there are massive implications for both the patient, for their families often, and our clinical colleagues who've got to counsel and manage that uncertainty, which can last for several weeks. And of course, from a health economics perspective, at a systems level, this is massively inefficient. So if you, next slide, please, thank you. So these are some of the steps that go from us receiving tissue all the way through to us receiving that final diagnosis in a form that's useful, in a form that's we can pass on to clinical colleagues at the MDT. And the next slide will show that it's not even as straightforward as that. Thank you. So we send our cases off, and just a couple of weeks ago, for example, I had a case where the report came back, and for one reason or another, the incorrect test had been requested. So we'd waited several weeks, and then we had to go back to send another request down to London for it to be analysed appropriately. Or we currently have a practice where we get the methylation array with the diagnosis, but then we often have to send it for subsequent NGS testing. And so that adds on a further delay rather than being able to do these things in parallel or indeed in one time. So what we really need, as the next slide will show, is a technology, a pathway, something that takes us from tissue to final diagnosis, the molecular diagnosis, as quickly as possible. And so we wondered in Nottingham whether there was a faster, a better way to do this. And if you flick through the next couple of slides, that brought us to about 18 months ago to a meeting with these two gentlemen. One on the left, Stuart Smith, who's an academic neurosurgeon here, and the key person, other person here is Matt Luce, who I think will talk to you now about the computational biology that he performs.
Hi there, yes, pleasure to be here today. If we can go to the next slide, please. Hopefully a video is going to start to play, but for those of you who are unfamiliar with how nanopore sequencing works, nanopore sequencing threads DNA through a small pore, and as it does so, you get a current change across the pore, which can be measured and decoded to give you the DNA sequence. And actually, you can do some really cool things if you can decide quickly enough whether you want to see that sequence or not. You can throw it away and capture another molecule. And this is something that we had been working on in Nottingham for some time prior to me meeting with Simon. One of the nice things about this technology is it's real time. It gives you the data straight away. So you don't have to wait for sequencing to finish to look at your data. If we can go to the next slide, please. So there's a number of different nanopore sequencers available, and this is showing the ones that we have run the work on that we'll be talking about for the remainder of this session, from the gridiron in the top left through to various different scales of nanopore device. They range from portable to very much not portable, but the thing they all share in common is this real-time data. If I could have the next slide, please. So what do you get from nanopore data, and in particular this nanopore adaptive sampling, choosing individual molecules as you go? Well, we've already heard from Patrick about the various different things you can get from whole genome sequencing. So single nucleotide variants, gene fusion events, changes in promoter methylation and copy number change, but also global methylation across the genome. So from that test directory, you can actually look at everything in one assay. So the methylation and copy number change can be diagnostic. They give you that methylation-based classification and the copy number variations that we would like to see. The promoter methylation on MGMT in particular is a prognostic indicator for treatment. And then there are potential therapeutic targetable mutations. And so when we first put this technology together, this adaptive sampling technology, we were very interested to see what we could do with it. And at that time, I was contacted, next slide, please, by a group in Heidelberg. Sorry, that's the one. And they got in touch, and they had been working for some time in a similar paradigm to the one we have here, perhaps slightly quicker. They were taking samples, they were running whole genome sequencing, they were running methylation arrays, they were running SNP calling, indels, fusions, all of those things, and running over a number of different tests over a period of days. And they thought that they might be able to accelerate this and use Nanopore for a single assay. And so we worked with them to develop that assay. And we were able to take all of the different tests they were running, put them onto a single sequencing flow cell. And we're showing here running for 3 to 4 days. And we were able to diagnose these tumours very quickly. That was published in 2022, and that was how I eventually met Simon, because he was very keen to see if we could do this here in Nottingham. Next slide, please. And so through some very generous support from Nottingham University Hospitals, we tested an initial 30 cases using this same workflow. Here, each case took about 3 days to run and analyze. We had excellent concordance between the Nanopore result and the standard of care results. There were some differences. They come down to various different issues, some sampling error, differences between the classifiers, and even some novel tumour entities that hadn't been described before. But overall, we were incredibly enthusiastic about what we had seen and what we found. Next slide, please. And so what we wanted to do was to see if we could move towards an even faster diagnosis time. And so on this slide here is Simon Deacon, who was a PhD student, and Swasti Kiani, who was a postdoc in my lab. And between them, they worked together to develop what we're calling an ultra-fast protocol. And this protocol allowed us to go from receipt of fresh tissue from the surgeon to a library ready for sequencing in about 90 minutes. And we could start to get these molecular classifications based on methylation and then ultimately molecular data, initial classifications in 2 hours and then final molecular diagnosis the next day. In order to do this, we've had to develop some software. And so if we could jump to the next slide. We have a piece of software that we've called Robin. Robin was named by Simon, and it stands for Rapid Nanopore Brain Intraoperative Classification. But because nanopore sequencing is real time, we are able to look and see what's happening in the data as the samples are in progress. And so in principle, we would be able to report to surgical colleagues or neuropathology colleagues the outcome of a tumour type, potentially even during an operation. And so at this point, I will hand back to Simon to tell you some of what we found.
Thanks, Matt. Yes, so after that initial retrospective cohort, we were really encouraged and wondered if we could do a prospective cohort in an intraoperative timescale. And so we've now done up to about 90 cases in total, and we've got very, very good concordance between standard of care and our novel pipeline. As Matt said, we have got some cases for which there is discordance, but there are good reasons why there is discordance. We haven't had any cases where we've got a different answer from our pipeline versus standard of care for reasons that we don't understand. It's really been very, very encouraging. So if you move to the next slide, what we'll show you now is we haven't unfortunately got time for the technicalities of all of this, but we'll show you some of the clinical impact that this might have. So this is an 11-month-old child's scan, and you can see a horrible looking brain tumor. And I did the intraoperative smear and found that it was just a very primitive tumor. It wasn't possible to come to a confident diagnosis, a very wide differential diagnosis of what sort of tumors it might be. And so as a result of that, the surgeons drew stumps after a subtotal resection because they didn't want to cause any harm for a tumor which they didn't know the biology of. And subsequently, this was found to be an ependymoma. Several weeks later, we got the standard of care, methylation array, etc., and were able to type it confidently. Unfortunately, this is a type of tumor that needs maximal surgical resection as soon as possible. And so this patient, unfortunately, had to undergo a second operation. Now, that's very unfortunate because, as you'll see in the next slide, we knew very quickly, and I think there's a video here, if you click on the, it's possible to play it. Thank you very much. What you'll see here is we use multiple classifiers based on the Nanopore data. Here we're showing you two, and you can see the diagnoses emerging very quickly. If you look at the Sturgeon classifier, the bottom one, within about 5 minutes of sequencing, we had a confident typing of the tumor that it was a particular type of ependymoma. And so the next slide will show you the morphology of it and the final diagnosis of this ZFTA-fused supratentorial ependymoma. We were able to deliver that diagnosis within an hour and a half or so of this tissue being received, and that would have impacted the patient's care, obviously. And so outside the intraoperative timescale, if we move to the next slide or two, what we really need to be doing is delivering these results, not when the system is ready to deliver them, but when our patients are ready for them. And so this is another pediatric case, a child here with a nasty looking tumor in the posterior fossa. And I did the intraoperative diagnosis on this again, and similarly had a differential diagnosis for it. And if you move through to the next slide, thank you. You can see again, this time, by this time we were using 3 classifiers. We had a very confident diagnosis, not just of the fact that it was medulloblastoma, but the molecular subtyping as well. Now, one of the great things about this software that Matt's developed in the Nanopore approach is that you really get all of the answers in one test. And so in addition to the diagnosis here, the next slide will show you that we get copy number changes as well. And you can zoom in on these and see that there are particular gains and losses, of course, all over the genome. This is a highly malignant tumor, but in particular, there is amplification in MYC. And this is an independent high risk factor for poor outcome in patients with medulloblastoma. Now, the next slide will show you the standard of care timeline. What we've been doing is all research only, and we haven't been able to share it with our clinical colleagues in real time. But this is the pathway for this patient who had an operation here. Their tissue was then sent to one of the national reference laboratories for this particular tumor. And it was 44 days before we were able to come to a final conclusion that we delivered to the MDT that this was a high-risk medulloblastoma on the basis of that MYC amplification. If we'd been using the Robin pipeline that we've described, this is what that patient's journey would have looked like. They would have had all the data available the next day, and as it happened, the operation was on Wednesday, our MDT is on a Friday. That MDT would have been able to have that. The MDT would have been able to have that information 43 days faster. So this is something we're looking very much to move into clinical practice as quickly and safely as we can. And the next slide will show you we held an education day 10 days ago or so now in Nottingham. We had, I think, somewhere near approaching 150 people come along, including Emma McCargo, you can see here, who's head of cancer for Genomics England. Next to her is the Promethean P2 integrated and the P2 solo, and we actually sequenced a couple of tumors, made the diagnosis in the lecture theatre, including doing the library prep, which you can see Simon Deacon performing there in the lecture theatre. And that's been written up in the latest edition of The Economist. And we're looking to work with people all over the place, including folk in New Zealand who've got a similar problem with a difficulty accessing molecular testing. So lots going on in this, and many people to thank. It's a real team effort. You can see a pretty horrendous photo of us there. But yeah, many people should be grateful for their collaboration with. Thank you.
Thank you very much, colleagues. Handing over to Prof Behjati, if that's okay.
And thank you for having me here. I will present to you this paper here, Benefits for Children with Suspected Cancer from Routine Whole Genome Sequencing, which is something we published, came out last week, if you're interested in sort of reading on the detail. And what I will talk about is essentially how we've been using whole genome sequencing in Cambridge, along with some data from our colleagues at Great Ormond Street, and report on whether or not we found this useful. Next slide, please. The first thing to say is it's a phenomenal team effort. So I haven't got time really to go for every single person here, but an enormous number of clinicians, lab scientists, pathologists, clinical geneticists. So it's a whole team, a whole crew who worked on this. There's a study that we conducted, as I said, across Cambridge and Great Ormond Street, and the data analysis was done here at Sanlam. And I say data analysis is a sort of hygiene, data hygiene that was required to make this publishable. Next slide, please. So the background is that it is, I think, reasonably obvious to state that whole genome sequence is a gold standard assay for reading a cancer genome. It is useful in pediatric cancer care in select patient groups, so there's quite a bit of evidence from different centers who used it in high-risk patients or children with relapsed disease, and to show that it actually makes a difference. And the reason for that probably is that genetic data does influence the treatment of many tumors. So you've just seen an example of how we use methylation classifiers to diagnose brain tumors. And in many, many different childhood cancer, we use DNA changes, DNA variants, mutations to guide treatment. And that's, I'm not an adult oncologist, but I suspect that's quite a huge difference between what we do in pediatrics and adult. In pediatrics, that's in leukemia. If you have mutation X, that means you need so much more intense treatment, for example. And the second thing is that the cancers that we treat are very varied. So it's a huge hodgepodge of different entities, some of which have no definition, and therefore it is likely, because a cancer is defined by its mutations, it is likely, therefore, that one would suspect that whole genome sequencing would be a useful thing to do. Now, what isn't clear, however, is whether, so we know, well, it's probably beneficial in high-risk cohorts, but is it actually useful if you offer it to every child who walks through the door who has suspected cancer? Next slide, please. And really, that's the question that we asked in our paper. Does routine whole genome sequencing improve the care of children with suspected cancer? Next slide. You're well familiar with this pipeline that has been set up nationally. This is how we do whole genome sequencing in the NHS today. It has grown on the back of the 100,000 Genome Project, which essentially set up the logistics for this. And it's complicated, but it works beautifully well. And so a child is diagnosed in hospital X. They then are not diagnosed. They pitch up an EDGH. Someone thinks they might have cancer. They then get sent to one of the 21 primary treatment centres for cancer, for children's cancer in the UK, where then typically a biopsy will be performed. DNA is extracted. This goes to Birmingham for plating. It then goes to Hingston, to the Illumina facility with which the NHS has a contract to do whole genome sequencing. They perform variant calling, and that data is then sent back to the GLH. We're now sort of in the middle box, and then the GLH scientists report it. They make that data available to the Genomics Tumor Advisory Board, where the data is discussed in the context of that specific child, and then a decision is made. And this bit is really quite critical. So if we look at other countries, the way it is typically done is that these whole genome sequencing childhood cancer whole genome sequencing efforts have arisen in academic units, and therefore all the discussion happens in one center, whether or not the patient is there. And the beauty of the system that we've got in this country is that actually the people treating the child get the data and can think about how that might be useful for their patient. It's a little bit like, let's say, if you had a scan, an MRI scan, you want to see that MRI scan when you have a discussion in your multiple disciplinary team meeting about the patient. You don't just want to receive a report, because you want to interact with it, you want to interrogate, you want to discuss it, and this is why it's so important to have that data available to the clinician who treats the child. And this, I think, is the beauty of our system. And the other is, it's just incredible that logistics exists to do this. It is just phenomenal. Next slide, please. Next slide, please. So what we did is the following. So about a couple of years ago, the NHS said, well, you can now offer whole genome sequencing to every child who walks through the door with suspected cancer. In London at Great Ormond Street, our colleagues started doing that in leukemia practice for every child who walks through the door. And in Cambridge, we've done it in our solid tumor practice. And therefore, we had two different cohorts, one leukemia, one solid tumors, in two different centers where we could ask the question, does it, if our aim is to offer it to everybody, and then we assess, has it influenced our decision making? So if you look under AIDS, that's the Great Ormond Street cohort, you can see there is the implementation phase, which is that initial bit from January to December 2021, and then from January 2022 onwards, we call that the routine phase, because that was the point at which it became routinely available. And what you can see on the left-hand side is, the point to labor is that essentially almost every family and child wish to have this, and it is doable in the overwhelming majority of children. If you then look at the right side, if you look at the children with solid tumors in Cambridge, again, it is essentially universally acceptable. One family decline, and when families do decline, it's a really good thing because it means they've got a very clear understanding of the issues related to it. It's normally about germline mutations, and they have a very clear sort of wish of what it is they want to do. And actually, I consider that to be a success because it means that the way we interact with families enables them, empowers them to make a decision that works for them. Now, also note that in solid tumor world, things are a bit trickier because obviously we normally want the biopsy of a child, but sometimes we don't because it's either unsafe or not indicated, or they have been biopsied elsewhere because of particular patient pathways. So an example is children with bone sarcomas, they wouldn't be biopsied in Cambridge, they would go to a sarcoma center for the biopsy, so we wouldn't have that sample. The unsafe category, so some children with high-grade brain tumors that sit, or low-grade brain tumors that sit in regions of the brain that cannot be biopsied, we wouldn't take a biopsy. And this is this sort of picture, and then some of them are also, I just highlight that category of Hodgkin's lymphoma, which isn't suitable for whole genome sequencing because there are very few cancer cells in a Hodgkin's lymphoma lymph node. And with that, we got this complete cohort of 282 tumors from 281 children. So for one child, we had 2 tumors. Next slide, please. And then we assessed utility. And the way we do that is we didn't retrospectively assess utility. We asked the question at the time when the data was digested in the MDT, what did the MDT feel, how the data was useful? And then we asked the question, were they justified in finding that useful? So they probably found a few more things useful than one might retrospectively think. And then we set together a bunch of us and discussed essentially every child together and reviewed what has been extracted from the data. And what you can see is there are sort of broad 2 categories. So there's this category of additional disease relevant findings. So these in about 1/3 of children. So these are things that are really interesting to know. So I found it really comforting. I find it very informative. I'm really happy that I know everything about the tumor of the child that I treat, but they're not things that one would say provide a clinical benefit. So for example, in the diagnostic category, we've had a few instances where the mutation makes a diagnosis that we didn't know before, but we had treated the child pragmatically anyway within that broad umbrella of cancer type that they're in. So it didn't actually change treatment, it just provided a label. That's important, that's interesting, but it doesn't change treatment. So we wouldn't count that, as it were. But then what we did count are these instances of clinical benefit. And the one that is the sort of the thing that we are after is the one where it changed management. And this is right at the bottom. So in 7% of children in 20 individual instances, whole genome sequencing provided a finding that changed clinical care. And the key thing about that finding is it was not delivered by any of the standard of care assays. And more importantly, no standard of care assay that one could have reasonably deployed in this child would have delivered it. So it's completely sort of off target, would have never thought this existed. And if we hadn't done whole genome sequencing, there's no version of reality where we would have done a test to look for that finding. Next slide, please. Next slide, please. So I'm going to give you one example because I think that helps. So we've listed in the paper in detail every single case in every single child in whom we think whole genome sequencing provided a finding that changed management, so that the reader can scrutinize this and decide whether they agree or not. But I'll give you an example here from our solid tumor practice in Cambridge. So this was a child, a girl who had a thoracic neuroblastoma, so a neuroblastoma in the chest. The neuroblastoma is a cancer derived from sort of nerves of the peripheral system. It normally arises in the adrenal, but it can grow along anywhere in the spine. And hers was in the chest, in the spine of the chest. And what we had to do is, as part of a treatment, we had to give a radiation, ionizing radiation. When we did whole genome sequencing, we discovered that she does carry a bona fide breast cancer predisposition mutation in a gene called PALB2. And that knowledge, so this is not something we would have ever looked at. So this is a really instructive example, because we can just briefly reflect on what tests we had done. So we do on a tumor, we do a copy number array in neuroblastoma, that's normal practice. We do direct sequencing of some genes that we are interested in. We had done FISH, so this is all standard practice. This particular family, because the mother was about to have another baby, she was quite worried about having a predisposition. So we also did a predisposition screen. So she had a copy number array on a germline done as well, and direct sequencing of a bunch of genes of the germline. But of course, that wouldn't include PALB2, because PALB2 is the breast cancer predisposition gene. One wouldn't look for that in a child with neuroblastoma, but we found this. And what this did in terms of changing our management, when we did give her radiation, I could tell my radiation oncologist about it, and then she had looked, Sam, I'm just going to reject the radiation field so that we do not irradiate her breast tissue. Next slide, please. Next slide, please. So there's then the huge question about health economical sort of considerations. And the bottom slide, next slide, please, of this particular slide is that across our cohort, we deployed 726 individual genetic assays that look for a genetic readout, be FISH, target sequencing, copy number arrays, and whole genome sequencing reproduced every single of those. We do not have the numbers from the NHS to be able to do a formal health economical analysis, but clearly that's what needs to be done. And it wouldn't surprise me if one could make whole genome sequencing cost effective and replace all tests that we currently deploy, all molecular tests, with whole genome sequencing. Next slide, please. Next slide, please. So, and then the other bit is, and that's what I tried to allude to at the beginning, in pediatrics, we come across unicorns very often and come across entities that we can't otherwise define. And the other beauty of this program is that it has helped us to find things we didn't know, and that might help children in the future. I could go for any of these here. If we want to go for the top example, we were able to find a disease-defining mutation of a particular type of benign tumor. And what that does is, now that we've got a definitive marker of this benign tumor, whenever pathologists aren't sure is this particular benign one or a malignant tumor, they can use this mutation to make that diagnosis. Next slide, please. So the conclusions from us are routine whole genome sequencing clearly can improve the care of children with suspected cancer, as implemented in the NHS in the 21st century, as implemented in Cambridge and Great Ormond Street and across England. This could potentially be cost effective, but that requires some detailed analysis and thought. And finally, it teaches us novel things we didn't know existed. Last slide, please. And with that, I wish to thank everybody here, the Wellcome Trust for providing my funding, of course, the children and their families. And I put up this picture here. The paper was featured last week, and Eddie very kindly told his patient story about whole genome sequencing. And the other thing about Eddie is why I'm very keen to put him up is he did the fundraising that funded the salary of the clinical fellow who conducted the study. Thank you very much for your attention.
Thank you very much, Sam. Now for the final session, which is a view from our partners, I'm happy to introduce Mike Ryan, Acting Medical Director, Head of Service, East Midlands Cancer Alliance, Amanda Pleven, Managing Director, East of England Cancer Alliance, and Gini Melesi, Head of Transformation and Lead for Clinical Networks, East of England Cancer Alliance. So welcome, colleagues.
Thanks, Emmanuel. I think I'll kick off, and just for clarity, head of service for East Midlands Cancer Alliance. So not medical and orientated, although I just wasn't smart enough at pre-med at university. So I'll give a bit of a generic update, if that's okay, in terms of where the East Midlands Cancer Alliance sits overall. And obviously, we have Amanda and Ginny from an East of England perspective. But before I get started, and I'll try and claw back some of the time because I'm conscious of it, I just wanted to say that I and East Midlands colleagues are very proud and privileged to be a part of what is an extremely exciting time and advances in not just technology, but knowledge and intel around how bodies work and the application of genomics and how that it applies to when it comes to cancer overall. The GMSA obviously doesn't just focus on cancer, but obviously that's our bread and butter. It extends to recognition of colleagues across not just EMCA, but also across our clinical leads, Vicky Kiesel and Umar Abdul-Majad, as well as colleagues across NUH, UHL, Cambridge, and the GMSA at that. And then finally, just a note further to the presentation earlier, as part of a case study from Simon and Matt, as part of the Radiate project under Stuart Smith at NUH at Nottingham. That's an incredibly exciting aspect within brain and generally in terms of nanotechnology, which I think is going to set a tone, which you may have recognized in more ways than one internationally. So thank you to colleagues for that. So I'll go ahead and start, if that's all right. Next slide, please, Ian. So generally speaking, what our Cancer Alliance is, I won't go through all the detail here because you can see and you'll see the slide backs, but effectively we're the primary vehicle for delivering the NHS long-term plan ambitions for cancer and improvements around cancer transformation. That's enabling business as usual services to get boosted and supported to transition into where we want to get to. Generally speaking, our early diagnosis rates, our outcomes data, as well as survivorship data is not where we want it to be. And so obviously investing in services financially, energy and resources to make sure we're increasing those rates for increased survivorship and outcomes is our main priority, while still improving cancer performance as a whole. Next slide, please. This has a few animations, so if you just sort of touch a few screens, just some key facts and statistics from a generic perspective. One of the things I hadn't realized previously is there's only one organ in the body that is not affected technically directly by cancer, and that's the heart. But when we break down a whole series of overarching statistics, it really basically summarizes the level of importance related to cancer, that it affects 50% of us at some point in our lives. And we're seeing increased numbers of diagnoses based on further work. So in effect, Cancer Alliances arguably are actually creating a lot more demand for all the right reasons. Workforce remains a key element of priority as part of that, not just in terms of investing and developing services, but also around how we work across primary and secondary care. And one of the things that we are emphasizing is how we don't just focus on early detection, early diagnosis, but better connect primary care clinicians with secondary care clinicians to ensure there's no differences or division, if you will, in terms of focusing on the patients and patient pathways. So long-term plan as well as a workforce plan remain key priorities. Next couple of tabs, if that's okay. One of the things that we have embedded as part of our clinical leadership model is to make sure that we recognize that genomics is helping us to understand what makes us tick and what makes us different, but also what makes us the same. And that's quite critical when you put into context Lynch syndrome alone, whereby 1 in 400 people are going to be walking around with Lynch syndrome in the country, statistically speaking. So when you think about England at 57 million people, that's roughly 140,000 people. And if only 5% of those know they have Lynch syndrome, that's 7,000 out of 142,000. And statistically, that's considerably daunting when you think about the opportunity at our foot. Next slide, please. Next slide, please. So obviously one of the things that we are focused on is trying to make sure that we add to the current premise of traditional delivery of services. So we also don't just think about symptoms, we also think about the risk that is part of that. Clinicians, as we know, are trained to be experts in identifying and managing risk, but that isn't always conducive or consistent with how the NHS and the systems work as a whole. So looking at how we define that, how we work as a whole between clinicians and non-clinicians, primary and secondary care, is something that we're focused on quite significantly, particularly when you put into perspective not just early detection and early diagnosis rates of on average 7 to 8% overall, but a good 90% of all referrals which come through the door into a specialist capacity on a 2-week wait, suspected cancer, will not get a diagnosis of cancer, may have a series of other incidental findings. Having said that, there's never no risk when it comes to this area, and so we need to make sure we're supporting. But also one of the main points is recognizing that whilst it can be a little bit daunting, a little bit challenging, the state of play and how we make progress can sometimes be very straightforward and sometimes not straightforward. Next slide, please. So across East Midlands, we've spent a huge amount of time over the past number of years, and some colleagues will recognise the painstaking element of some of that, but it's about understanding the broader impact of genomics when it comes to cancer across all steps of the pathway, from early diagnosis and even before that prevention through to those living with and beyond cancer, recognising that we're expecting over the next decade the number of people living with and beyond cancer is set to double. So making sure we understand the context of that in the round is critical. However, as part of this, we've established an expert clinical advisory group, which is multi-professional and also multidisciplinary to a certain degree in terms of how we function, which pulls in colleagues from all different disciplines and all different specialities to inform strategy. Next slide, please. So as part of that, we have an East Midlands wide overarching early detection and diagnosis strategy, which spans all different elements of the pathway, but also is conducive to bridging the workforce and educational needs and supporting long-term growth in terms of the intelligence and support mechanisms and enablers that are required. We've also worked quite closely with Emmanuel, Dick and colleagues to make sure that we're aligning some of that joint strategy, because of course on a national level, making sure we're connected and very clear in interpreting and applying the Genomics England strategy, the nice technology appraisals and overarching horizon scanning, but also some of the more advanced elements and programs related to GRAIL and CVLP, or the Cancer Vaccine Launchpad, is right up there in terms of how we're supporting with an infrastructure establishment. Next slide, please. I've only got a couple more slides overall. So more bread and butter stuff in terms of real practical things we're doing. Our role is really to connect networks, organizations, people, processes, labs, pathways. Not difficult there when you think about the number of players and enablers as a whole, but really to articulate and help support delivering a vision, which is strategic in nature. Collaboration can sometimes become a little bit overload. So we do work in different means and different ways to make sure that the delivery vehicle as the Alliance is genuinely connecting and moving forward. So we do quite a robust assessment and review and benefits realization approach. As I said, we've got an expert clinical advisory group established. We do a lot of horizon scanning and working with different colleagues up and down the country to better understand how we're working and feeding that back through, but also investing in not just the time and energy, but supporting colleagues through our primary care cancer training and education academy, bridging primary and secondary care, but also work with our local lab at Cambridge to and Nottingham, Leicester and colleagues to really support some of the conversations around diagnosis and the best treatment by turning around, improving turnaround times as best as possible. Ultimately, trying to make sure that we've got the right setup over time that will enable a tailored treatment and constant innovation as part of that. A lot of that is largely affiliated with tailored approach, but also dedicated clinical leadership that's supported to make sure the right thing for patients can become the easiest thing to do for clinicians. Next slide, please. Finally, just sort of scaling back a little bit in terms of where we are around East Midlands. I mentioned earlier in terms of outcomes and survivorship and why this agenda and the strategic imperative and criticality in terms of genomics, and thus the commitment is there, is that we have to improve outcomes and survivorship. We're below the power curve below average, if you will, nationally. And just doing what the national planning guidance expects us to do each year is just going to keep us across the East Midlands in that zone below average, which isn't good enough for any of us really when we put that in perspective. But when you look at the statistics, we've done quite a lot around health inequalities, health inequity, but also really generating around less survivable cancers, how we're going to get somewhere closer around early detection and early diagnosis. You can see there, as part of our commitment, is better understanding what the expectation and desired result looks like around the long-term plan versus where we're genuinely at since 2017 and more recently per tumor site. But also when you look to the right-hand side, you can see that typically the less survivable cancers have the highest volume of patients diagnosed at a late stage, which of course those top 6 will account for 40 to 50% of all cancer deaths. So the opportunity we've got is, and the effect of basically what we're trying to achieve over a period of time, is about reducing or improving lifetime, over the lifetime, improving mortality and morbidity for patients now into the future for our friends and family. Thank you.
Okay, so I'm Ginny Malaysia, Head of Transformation for the East of England Cancer Alliance, and I am a nurse, so saving the best to last. This is about the difference that nurses can make and what we're doing in the East of England. So why do we need to support our workforce to develop competencies in genomic literacy? And we've been hearing all morning how cancer is a disease of the genome. And so for healthcare professionals, it's really vital that they understand the importance and how it impacts on the work that they deliver. And I think the nurse's role is pivotal in this. Next slide, please. So with thanks to Graham, actually, who's one of our new nurse practitioners, and I have pinched this slide from him because I think it's great at showing how, there are, in the first instance, 1,205 reasons why we need to support our workforce to keep pace. And if you can keep pressing the buttons, it's actually got, I tried to take the delay off my copy, but I realised you've got a different copy. These are just the tests that are currently, as of January, that are available on the test directory, so encouraging clinicians to be able to perform these tests on their patients so that we can better direct their care. Next slide, please. And if we look at just lung cancer alone, and this is a slide around non-small cell lung cancer, adenocarcinoma is arguably our most common lung cancer that we see, and that's really a very important pathway for us to make more efficient. And you can see just how many tests, how many variants have been identified for this one tumour site alone. And all of these will impact on how that cancer will behave and will determine that the best treatment for that patient to have to be able to support their outcomes. Next slide, please. So actually, I would have, if I'd had the opportunity, put another slide in here. This is the last slide we had, the most up-to-date data on the number of patients receiving SACT, which is systemic anti-cancer treatments. And so that's a term now broadly used to encompass more than just chemotherapy, as we are delivering kind of targeted treatments and immunotherapies. And this shows that continual rise in patient numbers. And like I say, another slide would show in treatment numbers as well. And that's not only being driven by just us seeing and diagnosing more cancers, but it's being driven by the number of patients that are now having the opportunity to have other treatments that weren't available for them not so long ago. And in the very near future, probably early next year, there's another drug coming out for a large cohort of breast ladies that will impact on probably on a number of ladies or men that would not have gone into oncology previously, that they would have had their surgery and then not being followed up. But there's new treatments that, well, like I say, will be quite significant in number, will now be pushing that cohort of patients into oncology and having SACT treatment. And also just the number of lines of treatment now that are available, that once kind of your standard of care treatments have been exhausted and the patient's still well enough for treatment, there are now a number of other immunotherapy and targeted therapies that they can have. And so all the time, just the number of patients and treatments is increasing. Thank you. Next slide. Yeah. So this is a little tongue-in-cheek, but I think it just made me smile when I saw it, because it really shows that, you know, nurses are the workhorse of the NHS. And, you know, once upon a time we kind of made the beds and you know, and took temperatures, and over the years, and a number of years, just the role that the nurse has taken on has just kind of grown beyond all recognition. Next slide, please. So this is just probably to, it's a few examples of why nurses are key. The way that we diagnose, treat and care for patients and their families is constantly changing, and genomics cuts across all of this. And so I liked kind of the prediction, prevention, diagnosis and treatments, because it exemplifies some of the roles that nurses will have. And prediction alone, and much of this has been touched on this morning, but we can see now how tumours are driven, and that it determines how they're likely to behave to treatments and whether their cancers will even respond to treatments. And the nurse has a really important role in that, partly with a voice in the MDT. Just one example is the role that Oncotype, and there are other makes available, but now plays in a large number of patients with breast cancer to determine should they have chemotherapy or not. That once upon a time, you know, patients would be given chemotherapy if they were seen to be high risk, but that this test will predict better their response, and actually a number of high risk patients will now not have treatment, and similarly a number of patients that would have been low risk and wouldn't have had treatment will be given treatment now. So this is a really important thing. Prevention, and we've been a lot of the morning, and lots of, you know, stories from patients as well around that importance of understanding family predispositions to cancers, and that we can now diagnose those that have got a heritable origin and direct the care of not only the patient but their families, and, you know, the importance of risk reducing strategies and surgeries and screening. And with diagnosis, Brent's talked about, you know, a lot about ctDNA, and that's a really exciting thing that's coming through. Not only are we using circulating DNA and other liquid biopsies to diagnose cancers, but to monitor them for reoccurrence. And we've got a number of trusts now that are in the lung cancer ctDNA to improve the efficiencies on the path, the 62-day pathway. And we're encouraging some trusts to get involved in the colorectal ctDNA study that's looking at kind of after surgery, and patients need to have cancer vaccines, which leads us on to treatment and just that increased growth in targeted therapies, immune checkpoint inhibitors, which have arguably been around for a while, but the avalanche of kind of the onslaught of personalized anti-cancer vaccines. We've got melanoma out now, we're moving into colorectal, and I think the pace is really going to pick up. And, you know, nurses do have just such a role with teaching and communicating with their patients. They engage and they educate their colleagues, they speak up, hopefully, at MDTs, they're involved in educating their patients and the significance of testing and why it matters and what those results mean and what their follow-up care is. And they support patients in the worry that, you know, the huge worry here is, my cancer, if I passed it on to my children. Incorporating their genomic knowledge into clinical assessment, so identifying risk, and Europac is another thing. I don't think we've centered on that at all today, but that's looking at increased risk in families where there's a predisposition to pancreatic cancer, and just picking up from those conversations you have with patients that will inevitably reveal that there's a lot of cancer in their families, and that, you know, applies to Lynch and BRCA, as we've heard as well. And importantly, nurses are at the forefront of facilitating pathway changes, and there's a lot of pathway changing with these new treatments that are coming through. Next slide, please. So this slide is really just, I guess, just to illustrate a point. So cancer and genetics, again, as we've heard, go hand in hand. And 1 in 2 people now, according to the latest forecasts, will get cancer in their lifetime. And so it's really important, I think, that we know kind of bad luck versus genetics, and so many patients will tell us about the numbers of cancers in their family history. So how do we determine, and it's important that we determine, what is just bad luck, given that 1 in 2 people will get it, versus genetics, and that's an important question that I think nurses need to understand. Next slide. I think these graphs are somewhat the wrong way round. If you look at the one on the right first, it's it's looking at, I think this was done by Macmillan about 2 or 3 years ago, the nurses' use of genetic testing. So most of those, the first two tables on the right are about nursing accessing information for their patients or accessing tests for their patients. And only 3/4, roughly 3/4, just under, of nurses actually are doing that. But if you look conversely on this graph on the left, only a very small proportion, 34% of nurses, feel prepared for that use of genomics in their practice. So there's a clear disparity here between nurses ordering tests and informing patients, and that they actually have a really good understanding of that, despite 95% seeing that train is coming in the next 5 years. So knowledge and confidence really go hand in hand. So it's important that we give our workforce the confidence. Slide 10, please. I think we've only got a couple of slides left. I am aware of time. So how are we going to address this in the east of England? So Mike has talked about their strategy that they're following in East Midlands, which is our partner Cancer Alliance in the GMSA. And we looked at how we spent the Lynch funding that came down in 2023-24. We had already, over the kind of year to 18 months previously, done pretty good in getting Lynch syndrome testing across endometrial and colorectal cancers in as business as usual. We were still struggling, as is everywhere, with that mainstreaming, and that's very much down to just, that's another job for the CNSs to do. So how do we use that, it was quite a significant pot of money, to address and continue that sustainability of Lynch mainstreaming, but do much more than that? How do we get better value for our money? Next slide, please. So this is the team. There are a few honorary members there. We have Brent Carrigan, who spoke this morning. He is our clinical leadership kind of supervision for the genomics service, and Helen Hatcher, who is standing at the back there, who's the clinical lead for the GMSA and a consultant oncologist and TYA lead at Addenbrooke's. We then have Gloria, who's in front of Helen, and she was already in post and is the TYA nurse lead for embedding and improving uptake of whole genome sequencing across the East of England TYA ODM. And then the other members are the new team, and we have 2 lead genomic nurse practitioners. One is very much focused on kind of the Cambridge hub, and then we have Liz, who is more MSc. We have 4 or 5 trusts that face into London and Great Ormond Street hub, and then we have one nurse who was already employed by Milton Keynes, who faces into the Oxford hub. So we have a great team now. We have a person, a key worker within each ICB that have got good clinical supervision and 2 experienced leads. Both of them have got PGDs in genetics, and one's undertaking their masters. Next slide, please. So this penultimate slide. These are our programme priorities. We've had a face-to-face meeting. We had a really good discussion about the state of play that we were aware of across the region and how we wanted to focus. And training and education across primary care and secondary care is clearly key, along with mapping the gaps we can see in the service and the pathway improvements we want to do. Whole genome sequencing has come up very strongly, that there's a real lack of knowledge and a need to improve that about who we should even be doing that on. And TYA sarcomas, cut patients, and those that have had exhausted lines of treatment but are still probably appropriate to have active treatment. ctDNA, we want to support the continual rollout and making sure all patients have that opportunity. Europac, Lynch syndrome, BRCA testing for across prostate as well as breast and gynae, and that Jewish ancestry programme, and the cancer vaccine launchpad. So lots of work to do. Last slide, I think the next slide we're going to skip. So skip that one because Brent's done that to death. And my last slide is really what have we, progress to date. And we've had a great away day. We've had orientations with lead cancer nurses, and that's ongoing. And we're going to put some webinars on for our wider stakeholders to introduce it to the team and talk about what we're going to do and get your views on what we need to do as well. We've done a targeted survey, and the links are below. Now, I'm aware there are lots of surveys going on, and it's not to duplicate, but it's to really hone down on what we know as a Cancer Alliance and as a region we need to do, and what are the gaps in those areas and in the knowledge, and where do people feel they need our support. We've agreed our KPIs because, of course, we're going to need, this is a 2-year programme, and it would be good to have some sustainability at the end of it. I'm quite confident that you know, genomics will have continued to keep up there as kind of a top agenda item, and that there will be no doubt that these posts are needed, but hopefully that will be more embedded at trust level. So I think that is the last slide, other than just to say thank you. I don't think there's anything, no, and I don't think we've got much time for questions, but I'll hand back over to Emmanuel.
Thank you, Ginny. So Amanda will close the session. No pressure, Amanda, if we time-wise.
Of course, thank you very much, Emmanuel. And really, I just wanted to say a huge thank you. It's just extraordinary effort and passion and leadership from Ginny Malisi, who we're just so very lucky to have here in the east of England. It was the case that I think Mike gave a really good reference and recognition of the work of the Cancer Alliances, who work right across the country in the best interests of cancer patients. Now, we know how important this agenda is, how important genomics is, and as has been referenced many times today, we all have the pressures of not enough resourcing, whether that's time, people, money, but all with absolutely the same intention, and that is to do the very best for our patients. And I think the work that we've seen today, such as what is innovative for us in the east of England, is trying to work together to collaborate on things that can have the greatest impact and difference in a sustainable way. So we know for us there'll be some lessons learned, there'll be some things we won't get right, and there will absolutely be some things that we do, but we know just how important this agenda is. And so our intention in the East of England Cancer Alliance is to work alongside our clinical colleagues to enable you to do the very best that you do for the patients that you touch. So from me, really, I get the easy job, Emmanuel. It's a thank you to my colleagues. We will continue to share with you anything that comes out of the work in the East of England, as I know Mike will for Midlands, and together we'll learn to, I think, embed this service that is potentially so very impactful in how we meet both that long-term ambition of finding cancers earlier, moving from half to 3/4 that we want to do, but more importantly, almost the survivorship and the outcome for patients when cancer is confirmed, because that's where we have such an opportunity to do so very better. So thank you very much. That's all from me.
Thank you, Dick. And we would definitely value your feedback and would love to hear your thoughts on the event. So I think colleagues have posted a QR code in the chat, so please do give us your feedback and some of your thoughts. There are more sessions running, different themes, Wednesday and Thursday. Thank you, everyone, for attending, and thank you to the speakers as well.